Okay, did that work again?
Yes.
All right, thank you. So thank you everyone for joining. I'm Meghan Duffy from the University of Michigan, and I guess I am no longer officially the Vice President of the American Society of Naturalists, but I was for 2020. And so this is our delayed Vice Presidential Symposium, but I'm really excited that we're getting a chance to do it here in our virtual Asilomar meeting. And the theme for the symposium is model systems in ecology, evolutionary biology, and behavior. So I would say that a challenge we face as people interested in ecology, evolution, and behavior is that we're really interested in and fascinated by the amazing diversity that there is out there in natural systems. This is often what captured a lot of our interest and got us interested in the field. But at the same time, we're seeking to understand general phenomena and the processes and mechanisms underlying those phenomena. And indeed, the purpose statement for the American Society of Naturalists explicitly states that we seek the conceptual unification of the biological sciences. So I'd say there's a tension here, maybe a challenge. We love and celebrate life's diversity and all of its quirks, but at the same time, we seek to understand commonalities. And in my opinion, model systems can really help us to deal with this challenge. But I'll note that I think we only can do that if we have model systems plural, right? That S on the end of model systems is really important. And we need to have multiple model systems. We need to be thinking about different ways in which we can use existing ones. We need to be developing new model systems. And all throughout, we need to be using them thoughtfully and while aware of the limitations and strengths of different model systems. So I mentioned model systems plural being important. And one reason why I wanted to highlight this as a theme of the Vice Presidential Symposium is because developing new model systems or figuring out new ways to use existing ones is a real skill. It is hard. It is not something everyone can do. But I think it's really undervalued. I think people just sort of assume there will be a system for them to use for any question they might be interested in, and maybe sometimes miss all of the work and expertise and skill that went into establishing those systems or figuring out how to use them in a new way. And so our speakers today are all people who are doing really interesting work on model systems, often taking either existing ones and using them in new ways or establishing entirely new model systems. So to very briefly introduce each of our speakers, Nina Wale will challenge us to think about the use and underuse of model systems in studies of the ecology and evolution of infectious diseases. And just posting the preprint to Twitter led to all sorts of really exciting conversations. So there's definitely a lot to think about there. Nkrumah Grant will tell us about how he used a well-established model system, the long-term evolution experiment with E. coli, to ask about how relaxed selection influences fitness in other environments. So this very important long-standing evolutionary question. Carlos Garcia-Robledo will talk about work he's done in the rolled leaf beetle system that he's worked to develop. And he's using it, what he'll tell us about is how he's used it to study the resilience of insect populations to projected global warming. So it's certainly a very timely and important topic. And as an aside, I was so inspired by his system as I read through his manuscript that accompanies his talk here that I actually stopped partway through reading it and went and wrote an intro bio exam question about it. So I was super excited about his system. Next up, André Green will talk about developing the Monarch butterfly as an integrative Eco-Evo-Devo model. And I think this is a really great example of taking a system that was already well studied, but using it in really new and exciting ways. After André, Swanne Gordon will tell us about her work with what she describes as a non-model model system. I love that term. So the aposematic wood tiger moth, and she's used that to understand why populations show variation in warning signals. So you wouldn't expect that there should be variation, but there is. And she's used this system to help understand that. Finally, Rachel Penczykowski will tell us about how Plantago species can be used as models for studying both plant pathogen and plant herbivore interactions, and especially how it's well suited to doing that across environmental gradients. And I think the work that Rachel is doing with the Plantago system is really exciting and novel, and so I will forgive her for moving away from her first model system, which was Daphnia. Before we get to that, though, I want to note that there were so many more people who are doing really interesting work along these lines that easily would have fit into the theme of the symposium, and it was just so hard to choose. I started asking people for suggestions, and it was overwhelming. And so I wanted to feature just a few other people who I would have loved to have involved in this symposium, but I couldn't because of time constraints. So Amika Kamath, her dissertation work on anoles, to me is just like, it's amazing. And part of why it's amazing is it really challenged us to think about what we actually know about these model systems that we work with. Do we really know what we think we know? And showing us how much there is to learn, even with these really well-established model systems. Maria Rebegueda Gomez has worked on multiple different model systems, including yeast and mimulus, tackling really interesting and important and timely questions in ecology and evolution. Patrick Guerra is someone whose work I was not familiar with at all before I started asking people, and I was just so impressed. He uses two different systems, Monarch butterflies and also Cecropia silk moths. And he uses the latter system to understand how those moths spin cocoons that have really complex architecture. And finally, my colleague Louise Zaman uses a literal model system, self-replicating computer systems, to understand host-parasite coevolution. So as I said, there were just so many wonderful options of people doing this sort of work. And I wish we could have had, 15 people in the symposium, but that wouldn't have worked out. So I want to come back to this slide from earlier. As this poster says, life needs variation. And another thing that I wanted to emphasize in my introduction to the symposium is that science needs diversity. And our field has not made nearly enough progress in this area. And so I wanted to end by highlighting this new article on structural racism in STEM higher education, and to really encourage you all to read it. So I've underlined some of the, the whole thing is worth reading, but in the abstract, she notes, even at the top of the education hierarchy, Black STEM doctorate students and PhD degree holders consistently endure the racist residue of higher education institutions and STEM employers. And so she talks about these institutional diversity mentoring programs, and especially how they often seek to assimilate underrepresented students of color, and at the same time, they ignore or minimize the role or that the environments in which where we think we're inviting these underrepresented students of color actually can be really racially hostile work and educational spaces. And so she challenges us to actually tackle this, or else our aim to diversify STEM will continue at a snail's pace. And in her conclusion, so there are two paragraphs of the conclusion. This one I wanted to especially highlight, and especially this part in purple. So she says, what we really do not need in STEM is more of the same type of students from the same institutions, taught by the same professors, learning the same curriculum, working at STEM institutions where everybody looks and quite possibly thinks similarly. As long as there is widespread reluctance in these fields to address the insidious, complex effects of structural racism, which range from the individual to the institutional, STEM education will ultimately result in less robust, innovative, creative STEM industries and outputs. And at the end, she notes, especially if you've been looking at what's going on in the past week and thinking that you're in a little bubble where things are better, she reminds us that STEM is no exception to these larger societal patterns that we see. So again, I really encourage all of you to read this paper and think carefully about what you are doing and what you could be doing. Okay, so with that, I will move on to introducing Nina. is our first speaker. All right, need to click around here a little. So the first speaker in our symposium is Nina Wale, who will be challenging us to think about the use and underuse of model systems in studies of the ecology and evolution of infectious diseases. And Nina is a new assistant professor at Michigan State University. So Nina, I'll let you take it away.
All righty. Let me just share the screen here, 1 second. How's that?
That looks good.
All right, great. Okay, so thank you, Meg, for introducing the topic and for inviting me to talk among these wonderful, diverse young scientists. So Meg came to me and asked me to introduce the session with a paper or a talk that introduced the concept of model systems and why were they useful in ecology and evolution, specifically in the field that I might actually be qualified to talk about, which is infectious disease ecology and evolution. And my ostensible qualifications for this is that I have worked with a couple of model systems. I am trying to establish a new model systems with some strife, as Meg indicated, using Daphnia, which is really important in the ecology and evolution of infectious disease field, but as we know is a very important model in ecology and evolution in general. And I've also worked with a long established model of malaria, Plasmodium shabudi, which is important, again, both in ecology and evolution of infectious diseases and as a model in biomedicine. So ostensibly, I'm quite well qualified to talk about this, but it didn't take me very long to realize that I wasn't quite sure what I meant when I said the word model system. And indeed, that a few other people I talked to indicated that we sort of use the system in a very amorphous way. So it seems that I've fallen at the first hurdle. However, luckily enough, there is a wide body of literature in which what a model organism is, why they're useful, what their limitations are out there. So what I'm going to do today is I'm going to introduce you to one concept of a model system, a model organism that I've gleaned from this literature. And then I'm going to use that definition to ask, how are we using model systems in ecology and evolution of infectious diseases? And I'll be using the phrase EID as an acronym for this field. And then lastly, I'll go on to ask, well, if indeed we're not using them as models, and no spoiler here, I don't think we are, does this represent an underuse of these model systems? Okay, so what on earth is a model system? So the concept that I'm going to use today is one that is mostly based on the work of Rachel Ankini and Sabina Leonelli, whose work I really strongly suggest you look at. And they outline that model systems are a special subset of experimental systems that are firstly studied as representative of other organisms, such that we can study some or all of the fundamental processes and or phenomena that one's field is interested in. The second thing is that these organisms or systems are studied holistically. So we study everything from their cellular biology right up to their ecology. And in the context of ecology and evolutionary biology, then you might think that we study these systems from their genes right up to phenotypes to their impact on ecosystem processes. The last thing that this concept of model systems emphasizes is that these systems are studied in a comparative context. The idea is that we use them to learn about the rules of life, and thus we compare them, as Meg was saying, we compare model organisms between each other to find generalities about the drivers of the processes in which we're interested in. And as such, in order to do this, we need an infrastructure that allows organisms to be standardized and shared, and for resources and data to in turn be shared. So this might seem a little bit abstract at the moment, but actually it's quite familiar. Because if we turn to Joseph Travis's ASN presidential address in 2006, he noted that for robust inference in ecology and evolutionary biology, we need vertical integration, that is, to study multiple ecological and evolutionary processes in single systems. And then in turn, we need horizontal comparisons, the examination of these of the same processes in multiple systems. And so what Joseph Travis said really echoes at least the second and third requirements of model systems as defined by Ankini and Leonelli. Okay, so we have an idea of what a model system might be. Now let's ask, how are those organisms that we use in EID as models being used? Are they being used as model systems sensu stricto? So the first thing that a model system must do, as we've just learned, is that we must be able to address the fundamental phenomena in which we are interested with them, so that we can glean general insights about the field at hand. So in EID, what are those fundamental processes in which we're interested in? So ecologists and evolutionary biologists of infectious diseases are interested in the spread of pathogens among hosts, and indeed the evolution of hosts and pathogens. And similar to many ecological and evolutionary fields, we're lucky in that we have theory that actually tells us, formalizes mathematically what the processes that are really important as drivers of that central phenomena of interest, the spread and evolution of infectious diseases. Now, happily enough, weird phrasing perhaps, happily enough, we're living in a pandemic. So you guys already know this theory. You're very familiar with the fundamental, the theory that underlies the spread of infectious diseases. So we know that we have a susceptible host. Here's a little mouse, and it might get exposed to a coronavirus-like thing. And hence that host becomes an exposed host. After some time, that host becomes infectious. And this means that it can spread the pathogen to its friends and neighbors. Eventually, that host recovers. So this theory first describes the first key phenomena that we are interested in as ecologists and evolutionary biologists of infectious disease, that is transmission. And importantly, transmission is the measure of parasite fitness. So in order for a parasite to reproduce, it must go from one host to the next. So the second key phenomena in which we're interested in is disease. And this is the negative effect of infection on host fitness. And recovery, which is our third, is the amelioration of that negative effect. So we have three key phenomena in which we're interested: transmission, disease, and recovery. And by considering these, we can look at both the spread of infectious diseases and parasite and host fitness. Now, of course, there are many different parasite and host traits that contribute to each of these phenomena. So for example, there are different parasite and host traits that contribute to transmission. For example, in order for this little mouse to get this coronavirus-like thing, the parasite itself must have the required infectivity to be able to get in, and that host must actually be susceptible to that parasite. Similarly, disease is actually made up of both parasite and host traits. So virulence is often referred to as the parasite's contribution to disease, but there might also be a host component. So the host immune system might contribute via immunopathology. Lastly, recovery, according to the scheme that I set up, it consists of resistance, the capacity for the host to clear or kill parasites. Okay, so we now know that we've got three phenomena, transmission, disease, and recovery, that are central to EEID and must thus be addressed by our model systems. Now let's think about the scales of biological organization that are important to the ecology and evolution of infectious diseases, and thus must be addressed by our model systems, if indeed they are to be considered models. So variation, a variety of different biological scales of organization drive disease dynamics. So we might have interactions at the within host level, for example, competition between different parasites within the host. Similarly, there might be variation at an individual or genotype scale. So host genotypes might vary in their susceptibility, for example, while parasite strains might vary in their infectivity. Likewise, population processes are important. We know host density is very important to infectious disease dynamics. The spatial and temporal distribution of parasites might be important. And as we all know, host community composition might also be important, as might ecosystem processes. So variation of wide variety of biological scales might impact disease dynamics, and indeed they themselves might be impacted by disease dynamics, right? So we know that pathogens, for example, might alter the population density of a host. Okay, so we know what a model system in EEID must do. It must address the fundamental phenomena in which we're interested in, transmission, disease, and recovery, and it must address these different scales of biological organization that impact infectious disease dynamics. So what I did is I selected a number of model systems in EEID. These systems are to have a look at how we're using model systems in our field. These are widely considered as models in our field, and I chose them such that they covered the gamut of parasite and host taxa. So these systems cover a wide variety of parasite taxa, host taxa. They also vary in age. So we've got everything from infectious Escherichia virus, which was established as a model in the 50s, right to these three, which were established within the last 20 or 30 years. And what I did was I reviewed up to 20 papers in each of these model systems. And what I said was, since model systems are a special subset of experimental systems, each of these papers had to include an experiment. And specifically, I said that it had to include an experiment in which an infection was initiated, since that seemed to me like the most basic experiment that one had to be able to achieve in order for a model to be, a system to be considered a model experimental system in EEID. And note that these studies could include a experiment where an infection was initiated in a mouse, for example, and the infection followed, but it also covered studies where somebody went out into the field, brought the parasites back to the lab and initiated an infection just to phenotype those parasites. Okay, so what are we doing? How are we using our model systems in EEID? Let's first ask, are we addressing the fundamental phenomena that we've identified in our field with these systems? So here, I've got 3 boxes to represent each of these phenomena, and the hotter the color, the higher the percentage of studies that addressed these different phenomena. Note that if a study focused on both a transmission process and a recovery process, then they could be allotted to both boxes. And just a note of how I did this, for each of the papers, what I did was I actually noted which of the different sub-processes that are involved in transmission, disease, and recovery was being studied. And then I counted up which of the sub-processes contributed to each of these umbrella processes. So what you see is that we're studying transmission and disease quite a lot, in a high percentage of studies in all of the systems, but we're not really studying recovery very much. So it seems that we've fallen at the first hurdle. We're not necessarily even looking at one of the key processes in which we're interested in. So why is this? So one reason for this absence of studies in recovery is that it could just be the way that I did this, right? I was looking for papers that looked at resistance, that's the killing or clearance of parasites from the host. And it was only resistance that I considered to be a recovery process. Now we know that, for example, as Dan Bolnick pointed out to me, tissue regeneration could also be considered a recovery process. But that's not what I think is going on here. First of all, I didn't find a single study that was looking at tissue regeneration. And second of all, it looks like we actually aren't giving ourselves even the chance to look at recovery. Since full recovery, that is the complete clearance of parasites from the host, isn't even possible in many of our model systems. In 4 of the 10 model systems, hosts don't recover at all. And in 2, it's very, very rare. So rare that this information was gleaned from a DM on Twitter. So it doesn't look like we're studying one of the 3 key phenomena in EID with our model systems. And in fact, the picture, I'm afraid, gets even worse, because when you start to look at the sub-processes that make up these 3 phenomena, the picture looks even worse. So it does look, as I said, it does look like we're studying disease quite a lot. But when I was actually categorizing these studies, I found it really difficult to say whether a study was, for example, looking at virulence versus immunopathology. And instead, I just had to say, oh, well, the experimenter is looking at morbidity, that is just sickness. And this occurred in the vast majority of studies of disease. It wasn't clear that there wasn't the variation at the host or the parasite level to delineate whether we were studying the parasite contribution to disease, that is virulence, or the host contribution to disease, that is immunopathology. And this is a problem because if we want to study host-parasite coevolution, then we need to be able to delineate the different contributions of host versus parasite to disease. The second thing that was quite interesting was that though we are studying transmission and the processes that actually that join up to contribute to transmission quite a lot, we're actually only studying a few of them. We're studying infectivity and replication of parasites quite a lot, and we're studying susceptibility of hosts quite a lot. However, we're not studying this realized transmission or what I've called entry to exit very much. So what on earth am I talking about? Well, I called I said a study was looking at realized transmission if it looked at an infection from its initiation in a susceptible host right through to its transmission to a second host. I said a study belonged to this entry to exit category if it studied an infection from its initiation in the susceptible host to its exit from the host. And as you see, we're not doing this very much, and we're only doing it in a subset of those 10 systems. And again, I think this is because we simply can't do this with our model systems. So in 2 of 10 of the systems that I looked at, we cannot complete the life cycle of the pathogen in the laboratory. So there's no hope of studying realized transmission. But when you take a deeper look, the picture actually looks worse than this. From personal experience, I can tell you that completing the transmission cycle of Plasmodium schaeberti in the lab is really, really tough. Similarly, secondly, IEV, this old system, is no longer even in use as a model of ecology and evolutionary dynamics. And that's a real shame, because this was one of the systems where there were epidemic studies done. So in 40% of our model systems, we can't really even study transmission properly. So it looks like at the level of fundamental phenomena, we're not doing a fab job. We're not necessarily studying the processes in which we're interested. So what about scales of biological organization? Are we looking at those, the processes, the different scales of biological organization that impact infectious disease dynamics? So disease dynamics, recall, are affected by variation in parasite and host traits at each variety of different biological scales. And what I did with each of the studies is I asked, at what scale was the independent variable in the study, and what scale of biological organization was the response? So for example, a study where the investigator was looking at the effect of host genotype variation on the outcome of population interactions between parasites within the host would be assigned to this category here. And since it's since there was only one study in this example, it would this block would be bright red, right? 100% of studies were in this category. So recall that while we're interested in the scales of biological organization that are being studied, is because we're interested in vertically integrating our model systems, looking at a variety of processes at different scales to get a holistic impression of infectious disease dynamics. So if a system was fully vertically integrated, you'd expect this box to have some color in each of these different squares. So the whole panel would be filled in. Unfortunately, however, it doesn't look like we are quite achieving vertical integration of our systems. So as you will see, we have these panels, each of which represents a different model system, are filled in a lot of the time on the top left corner. That is, we're studying the interactions between processes at scales at the individual scale a lot, and their impact on within host dynamics, for example. However, it's very rare that we're studying how within host individual level variation impacts parasite population parameters, host population parameters, epidemics, communities, etc. We're doing that very rarely. Indeed, and lastly, we're very rarely looking at the impact of ecosystem dynamics on parasite processes and vice versa, right? This part of the graph is almost always white. So it seems that the model systems that we have in EID might not really be models at all, right? We're simply using them, they're not to study very specific phenomena and processes at only a few of the biological scales that matter. So does this represent an underuse of our model systems? So the first thing I want to say is that I'm going to highlight two underuses, and in the interest of time, I might cut this. The first underuse was actually revealed by doing this very exercise. And it's an underuse that I think could be easily remedied. And if it was remedied, it could give us a lot of information about the ecology and evolution of infectious diseases. And it relates to this third requirement of model systems, that they're studied in an explicitly comparative context and there's an infrastructure to do so. And note that when Travis gave his presidential paper, he noted that these horizontal comparisons were really powerful when they were conducted quantitatively. Now, what I showed you was that we are underusing our systems, our model systems, and that we're only looking at a few of the processes that matter. Ironically, this means that there's actually a great opportunity to do horizontal comparisons within our field. We have a lot of data on a few processes, and that means that there's an opportunity to integrate that data to ask, are there general rules that work between systems to explain the processes in which we're interested in?
So 4 minutes left, Nina.
Okay, I'm going to wrap this up quickly. But one of the things I will say is that I don't think we quite have the infrastructure to do this powerfully and quantitatively yet. I looked at many papers and I saw that we don't have concrete definitions of the processes in which we're interested in, and that will thwart powerful quantitative conclusions. And moreover, we don't have a really good infrastructure for conducting experiments that are designed consistently across fields that will help us to make horizontal comparisons easily. The second and last thing that I'm going to highlight is this transmission gap. The fact that we don't necessarily study the realized transmission, the whole infection process from initiation to transmission in our experimental systems. And of course, this fundamentally means that we cannot vertically integrate EID. We cannot start to study the interactions between processes at the individual level with those at the epidemic level and hence community and ecosystem level. And this is important because it is very difficult to study such cross-scale interactions in the field. And this is where model systems can be really powerful. And in the interest of time, apparently I'm slowed down over Zoom. I'm going to end there, but I'd be very happy to discuss why we have such gaps and how we might fill them going forward. Thanks very much. Sorry, you missed all this great stuff.
Well, thank you very much, Nina. And at this time, if anyone has any questions for Nina about that very interesting talk, you can put them in the chat or you can put them in Slack. I'll be monitoring there. And while people are starting with their first questions, I have quite a few, so I'll throw some at you.
Great.
I'm interested in, it looks like what you've done here is kind of a meta-analysis of specifically, studies of model systems, mm-hmm. and whether that kind of systematic approach needs, has some predicate to it, like a well-developed theory that allows you to pinpoint the kind of important processes in disease transmission. So if you have, say, an emerging field where you're using model systems to get your way and you don't have a well-developed theory,
yeah.
Can you do this kind of process and ask where are the gaps?
So I'm really pleased that you raised this idea of theory. So I actually think I'm going to do a politician on you and sort of switch your question. I think in ecology and evolution, we're really well set up to do this sort of analysis because we have theory, right? I have Anderson and May's theory that describes transmission, and that means that we can identify the processes that are important at hand. You could do the same in population genetics that has established theory, et cetera, et cetera. In an emerging field, of course, I think that that's going to be really difficult. But I think that in ecology and evolution, even if you're trying to take a new model system in a new direction, you know the fundamental principles that underlie ecology and evolutionary biology, right? So you could sit back and say, all right, I need variation, I need I need some set amount of variation in this process that I'm interested in, et cetera, et cetera. So I think even from first principles, you could sit back and say, what do I want from a model system? And then the other thing to do, as I would say, is as your new field is developing, doing this sort of exercise would, as you go, would be quite instructive. So one of the things that the great joys of this exercise was to look back at the history of my field and ask, well, are we meeting our priorities and how have we met them historically? And actually, as you'll see in the paper, and I wish I'd been able to get to it, there are sort of historical contingencies that happen. We lose a system or we lose a trend because it goes unfashionable, or somebody dies, or actually, in one case, the Homeland Security didn't let a system into the border, and so we just stopped studying it when a researcher came into the country. So I would say that if you do this sort of exercise, either guided by first principles, be they verbal or embodied in theory, through as your field developed, then it could help you to identify gaps.
I think we're at time with that. So there are more questions, but Nina could potentially answer them in the chat or
yes, I could. I'll sit here and do it in the chat.
Great.
Thank you so much. Great, bye.
So next up is Nkrumah Grant, who is a postdoc at the University of Idaho, who will be telling us about using E. coli as a model organism for evolution and physiology in order to understand how you maintain an ancient metabolism. And I promised you there'd be My Little Pony in the background, and there is. All right, so I'll turn it over to Nkrumah now.
First and foremost, I would like to thank the organizers of this seminar for organizing what has been an amazing conference so far. This is my first ASN meeting, and I must say that it is a privilege for me to contribute a talk to this symposium. My name is Nkrumah Grant, and I'm a postdoctoral fellow at the University of Idaho. And today I'm going to speak on some of the work I completed during my dissertation studies at Michigan State University, where I utilized Escherichia coli as a model organism for evolution and physiology to understand the unselected maintenance of an ancient metabolism. Now, since this is a session on model organisms, I first want to start my talk with an operational definition for what a model organism is. And I define this as an organism whose genetics, physiology, habitat, and behavior are reasonably well understood, such that these variables can be manipulated and the insights gained from such manipulations applied more generally across the diversity of life. Now, some common model organisms used by research scientists includes your Brewer's release, Saccharomyces cerevisiae, your common house mouse, Mus maculus, and the nagging gnat, Drosophila melanogaster. And I'm sorry if there are any Drosophila scientists in the audience today. They tend to bug me, no pun intended. Now, all of these organisms share features that make them good model organisms. They're easily reared in the lab or in the field. They've reproduced very rapidly and have relatively simple and understood genetics, allowing for changes to be introduced, isolated, and then studied, right? Now, while these have been good model organisms, the best studied and most utilized model organism to date was discovered by the one Theodor Escherichia in 1884, a German microbiologist and pediatric disease physician. Now, during a routine study of neonates, he found that there was a common rod-shaped bacterium that readily colonized the guts of neonates in as early as 3 hours after birth. Now, he originally named this microbe Bacterium coli commune, but which has now become known as Escherichia coli. And E. coli is a model organism, a good model organism, or an ideal model organism for several reasons. It's small, so it doesn't take up much of that lab space. Most strains are non-pathogenic. It has simple nutritional requirements, which make it readily culturable. It's asexual and can divide in as little as 23 minutes, given the right growth conditions. Its simple haploid genome removes the complications of zygosity. It evolves very rapidly, approximately 10 to the negative 10 mutations per genome per replication. It could be stored long-term and revived with minimal loss of viability. And it is ubiquitously found in most warm-blooded mammals and often in birds and reptiles. And it is for these reasons that during the molecular revolution of the 1950s, that eyes were set on using E. coli to understand general biological phenomena. Now, in the last 30 years or so, these features have also established E. coli as a model organism for studying evolution. Now, this was done by the one Richard E. Lenski, an evolutionary biologist and my dissertation advisor, who was interested in asking a few simple questions with respect to evolution. He wanted to know what are the dynamics of evolution, including the rate of change. How repeatable is evolution? And he also wanted to know what more we could learn about the relationship between the genotype and phenotype. And to answer these questions, he started a relatively simple experiment that would go on to create an entirely new research field, that is experimental evolution. Now, the experiment, and similar to other experimental evolution studies, is simple in design. In this case, 12 populations of E. coli were started from a single ancestor and grown in a glucose-limited minimal media, where they divide approximately 6.6 generations per day before reaching stationary phase. Now, every 24 hours, 1% of that stationary phase culture is transferred into fresh growth media, and this has been repeated over time, such that by our current year, 2021, the populations have been evolving now for greater than 70,000 generations. Our E. coli strains have no active prophages, there's no horizontal gene transfer, and there's no sex, which isolates the effects of mutation, natural selection, and genetic drift. Moreover, because these are bacteria, they can be frozen and revived, and whole population samples and clones from each population have been frozen every 500 generations or 75 days. Now, this has been useful because where other studies have been, where a lot of evolution and historically have been, inferences have been made using incomplete fossil records, this creates a frozen fossil records from which then we can go back retrospectively and then assess the evolved and ancestral strains in adaptive and non-adaptive environments. And using this simple evolutionary marker, we can discern the each of the derived and the ancestral forms just by virtue of mixing them and plating them on media that allows one to form white colonies and the other to form red. Now, many adaptations have occurred in this experiment, highlighting the repeatability and randomness of evolution. For example, evolved lineages are more fit than the ancestor. Fitness increases rapidly in the beginning of the experiment as mutations with highest benefit are fixed in populations, then slowing down as the experiment proceeds. Each of the 12 lineages has also increased in cell size. So here I'm just showing you the ancestor and a representative representative population from this experiment. And on the random side of the coin, half of the populations have evolved hypermutability, such that the average hypermutated population, as we call them, has upwards of 1,000 mutations, whereas the non-mutated populations have on average 70 mutations. But more importantly, and the most striking adaptation that has evolved in this system is the evolution of aerobic citrate metabolism. So the minimal glucose media that these populations have evolved in also contains citrate as a chelator for iron. And this one single population evolved the ability to utilize this carbon source for growth and reproduction. So the LTEE then has been a great system to study some of those initial questions that Professor Lenski initially sought to answer. However, while the 12 E. coli lineages are adapting to the LTEE environment, E. coli's natural habitat is not the flask. This then also makes the LTEE a good model system to study the effects of relaxed selection. Now, this is a term that many may be unfamiliar with, but what is relaxed selection? Relaxed selection can be defined as the weakening or complete removal of selection on a trait. And relaxed selection predicts trait loss. Take here, for example, this cavefish, Oceanus moxicanus, that is derived into a surface-dwelling form and a cave-dwelling form that lacks pigment in its skin and eyes altogether, because who needs to see in the dark? Now, these losses are characterized as what we as evolutionary biologists call trade-offs, and are primarily the result of two processes. Antagonistic pleiotropy, where selection for one trait comes at the expense of another, and mutation accumulation, where genes that are not being used in the environment accumulate mutations because selection is not actively selecting the best mutations or removing the worst ones. Now, the negative effects of such evolutionary trade-offs are only observed when the selection pressures are reintroduced. So in the cave, the blind cavefish will do well. But if you were to put that variant in an environment where it had to evade predators or actually search for its food, it wouldn't do so well. So we can say that relaxed selection can be summed up generally with one simple idiom: you don't use it, you lose it. But I challenged this with another question during my dissertation work: or do you? And I approached this from an understanding of pleiotropy and from an understanding of genetic networks. And I wanted to know whether or not traits with more genetic interactions or those with deep evolutionary relevance, if they would be safe. Such that here, as I'm showing you in this E. coli gene regulatory network, to put that into context, are genes that have less partners or that are on the periphery of a network more likely to be lost than genes that are very well integrated. Now, to answer this question, I thought about many of the traits that could have been under relaxed selection in the LTEE. And what dawned on me was that because E. coli is a facultative anaerobe, meaning that it could grow in the presence or absence of oxygen, which I call metabolic plasticity, anaerobic metabolism has been under relaxed selection. And this is because the flasks are constantly aerated in the LTEE, for one, and for two, the growth medium does not support a high enough population density such that the cells would grow to some mass that would lower the oxygen concentration in the flasks. So I used this then, and a basic question of my dissertation research, at least chapter 1 of that, was: is metabolic plasticity retained? And I wanted to know whether or not I can understand this from a genomic and phenotypic bases of the trait. Now, given the predictions of relaxed selection, there were some naive hypotheses. For one, from the genomic side of evolution, we would expect to see a higher abundance of mutations in anaerobic specific genes when compared to those in aerobic specific genes, and this would be characteristic of relaxed selection. Now, on the phenotypic side of the coin, we would expect to see stunning performance of evolved lineages in an anoxic environment when compared to the ancestor or earlier descendants. Now, I will say that while I did have some expectations for the genomic side, and I did believe that this would occur, when I was considering the phenotypic side of the coin, because I know that anaerobic metabolism evolved 2.4 billion years prior to the evolution of aerobic metabolism, I thought that this deep evolutionary relevance would have selected for the maintenance of this trait over time, because as you know, life proceeds kind of like a scaffolding type, by scaffolding, things build on top of each other. And I felt that in my mind, that getting rid of the anaerobic metabolism would have too many consequences for things that were built on top of this network. Now, to answer these questions, I use publicly available data sets for the genomic questions. And what I used was metagenomic sequence data that was collected by Ben Good and colleagues in 2017. And these metagenomic sequence data, they include sequence data for populations that would be for cells that would eventually be the winners, as well as those that acquire mutations off the line of descent. I also utilized information available for E. coli as a model for physiology, its long-lasting history as a model for physiology, and I identified 227 and 345 genes that were differentially expressed with a 99% posterior probability in oxic and anoxic environments, respectively, And I call these my aerobic and anaerobic specific genes. And I basically then use this information to then query the metagenomic sequence data to ask, what is the mutation frequency in these genes? From the phenotypic perspective, I basically used those competition assays. I grew the ancestor and evolved lineages in co-culture in anoxic and anoxic environments, and established the winning strain by counting the colony forming units for each strain, again, based on the colors of their colonies. And this then allowed me to establish the relative fitness of evolved and ancestral strains. Now here I am showing you a metagenomic sequence plot. Each panel was a population, the x-axis time and generations, and the y-axis allele frequency. Now you might notice that some of the plots look messier than others, and this just reflects those populations that have evolved the hypermutator phenotype. And what I wanted to do then, I wanted to look at the stars. So I wanted to count the number of stars, which just shows the appearance time of the mutations. And I wanted to know whether or not in these aerobic and anaerobic specific gene sets, the frequency that those mutations popped up. So this test here, I'm just looking at 50,000 generations. And here I'm showing you in the hypermutator populations that there are more mutations in the anaerobic specific gene set than in the aerobic specific gene set. And this is characteristic of the relaxed selection that we expected to see. Now there's a different expectation for the non-mutator populations. And this because the non-mutator populations, they don't see a lot of mutations during their evolution. And because of that, selection is only picking out the better ones. So we would expect then the opposite trend, where we would expect to see the aerobic specific gene set accumulate more mutations than the anaerobic specific gene sets. And that's exactly what we see. And this indicated a good sign and signal of positive selection. Now, it was this clear contrast between the hypermutator and non-mutator populations suggested that if we could compare the mutation frequencies across time in some type of way, we can get a better understanding of the selection dynamics in these systems. So working with my colleague and collaborator, Dr. Rohan Madamseti, we developed a method that would allow us to make this comparison. And this idea was very simple. The thought was that we can randomly sample N mutations within each of the genomes, where N has the same cardinality of our gene sets of interest. So in this case, an anaerobic specific gene set or an aerobic specific gene sets. And then we can integrate those mutations over time to generate a null distribution. We can then take the mutations that we observe within these populations and compare it to this null distribution. And if those observed mutations fall into the obscured or into this null distribution, we would say that selection is obscured. If it fell below this null distribution, we can say that was evidence of purifying selection. And then we can have evidence of positive selection and a combination thereof if you fall above or you start to change as time proceeds, right? So on the next slide, I'm going to show you the results of these analyses when we applied them to the aerobic and anaerobic specific gene sets in the system. The aerobic specific genes are going to be shown in black, and the anaerobic specific genes are going to be shown in red. So here what I'm showing you is the generation time here on the x-axis, and on the y-axis, I'm showing you the cumulative number of mutations normalized by gene length. And what we can see in the non-mutated populations is exactly what we expected, a strong signal of selection on aerobic specific genes. These black curves fall above this gray null distribution. Now, it was more nuanced in the hypermutated populations. We see that our two gene sets lie in this obscured region that I described earlier. Now, this is expected because of emergent neutrality, where both beneficial and deleterious mutations fixed at nearly mutual rate. There are a lot of them. However, if we compare the curves of the red and black curves, these mutation frequencies, we see that more often than not, the red curves fall above the black curves, suggesting that there's more mutations that are accumulating in the anaerobic specific gene sets. And again, that's reflective of relaxed selection over time. Now, interestingly, using our method, we was also able to find signatures of purifying selection in two lineages, error +3 and error +6. In the aerobic specific gene sets, both accumulated mutations in the aerobic specific gene sets that were lower than the expected null distribution. Now, I will say that oftentimes in evolution experiments, or when these hypermutator populations evolve, and this is true for Pseudomonas, also true for Bacillus subtilis, and also in some cancer lines, these hypermutator phenotypes are often ignored. You can't glean anything or any information from an organism that has thousands of mutations. What is doing what and where is it doing it, right? But what I'm showing you here with this method is that we were able to resolve using these hypermutator populations and this method, this statistical method, we were able to resolve positive selection, relaxed selection, and also purifying selection. Now, after focusing globally on all of the mutations, I wanted to center my attention on the genes responsible for the aerobic to anaerobic switch, the fumarate and nitrate reductase protein, which responds to oxygen, and the anoxic regulatory control system, which is a two-component regulatory system that responds to the redox state of the cells. Now, when we look at the FNR protein, we see that only two populations get a mutation in those genes, and then it drops out. So they're at 30,000 generations, and I think by 35,000 generations it's gone. However, when we look at the mutation frequency in ARC-A and ARC-B across the lineages over 50,000 generations, we see that all populations have a mutation in either ARC-A or ARC-B. And in fact, ARC-A is one of the top 12 genes under selection in the long-term evolution experiment. Moreover, these mutations, they fall in domains relevant to protein function. Here I'm showing you that in this bottom figure here. Now, it is well known that ARC-A and ARC-B signaling mutants are sensitive to photosynthesizing dyes, and that's because these dyes increase internal ROS concentrations. So this can be used then as a test to determine whether or not signaling in these strains are not well. So I plated our populations on dyes on toluidine blue O. Here I'm just showing you an ARC-A knockout, the ancestor in ARC-B knockout. And I'm showing you here each of the populations at 2,000, 10,000, and 50,000 generations. And what we're looking for is a sensitive phenotype if ARC-A signaling has been affected, where you don't form a confluent spot like the ancestor, you have these punctuated dots. Now at 2,000 generations, none of the populations had mutations in ARC-A or ARC-B, and all of them seem to do well on this media. At 10,000 generations, 75% of strains that have ARC-A mutations and 67% of strains that have ARC-B mutations are sensitive to this dye treatment. You can see that here with these lack of confluency. And at 50,000 generations, 86% of ARC-A mutants and all of the ARC-B mutants are sensitive to this treatment. Some didn't even grow at all. Now, if you know anything about the system, these populations have been evolving in the minimal media, as I said earlier, so it's relaxed selection for other growth substrates. So one can ask whether or not this is a dye phenotype or if this is just a media effect. So by removing the dye, and as you can see, just by removing the dye, I was able to rescue sensitivity in all strains, but except maybe two. Now this suggests then that ARC-A-B signaling has been altered and implies that evolved populations are highly susceptible to oxidative stress. But I wanted to know what the fitness of these evolved populations are, of these evolved clones were in an anoxic environment. And to do that again, I just used those competition assays. Now we would expect for the evolved clones to be better adapted to the environment that they evolved in. And that's exactly what we see. The fitness measures fall below the isocline, demarcating the two environments in favor of growth in the adaptive oxic environment. Now we can then take this information and then we could calculate Grant means and look at the overall fitness trajectories across time, and we find the following. As expected in the oxic environment showed here in black, fitness increases such that by 50,000 generations, the average population is about 55% higher than that of the ancestor. Now, interestingly, fitness also increased on average in the anoxic environment, here shown in red, through 10,000 generations. It was significant. And even so at 50,000 generations, while we didn't see any more increases, this fitness was still at a level that was comparable to the ancestor. And again, we wouldn't expect that because these environments, these strains have not seen this environment, right? And again, relaxed selection predicts trait losses. So we'd expect for them not to be well. So this suggests that anaerobic metabolism is buffered against the effects of deleterious mutations in some type of way. So after looking at this data, I wanted to know whether or not there was evidence that the aerobic and anaerobic specific gene sets were connected in any type of way. And to answer this question, I used a database called String, which is just a database of protein association networks. And here is a figure from that analysis. Here I'm just showing you again the anaerobic specific genes in red and the aerobic specific genes in black. Now, as a reminder, we identified these genes as being 99% differentially expressed with 99% posterior probability in the aerobic or anaerobic environment. And here we're showing that they're much more integrated. They're much more connected in some type of way. What is the relevance of these connections remains to be tested. Now, this to me sheds light for a hypothesis that was proposed by David Lappy called buttressing pleiotropy. And this buttressing pleiotropy, the idea behind this is that the function of an unselected trait can be maintained by selection acting on another trait it is correlated with. And this has not been observed in biological systems to date. However, I believe that anaerobic metabolism and its long-standing history as its 2.4 billion evolution years of evolution before the anaerobic metabolism evolved, right, sheds light that yes, these connections may actually be relevant and important for the maintenance of this trait. And I think then too that ARC-A and ARC-B may be the key to this regulation, but that also remains to be tested. But I do have some initial data that shows that that may be the case as well. That being said, I'd like to conclude with a couple of statements. I'd like to say that evolution experiments, while they are ideal models for understanding evolutionary processes and positive selection, they're also useful for understanding the effects of relaxed selection. And I think this is an underappreciated characteristic or feature of evolution experiment. I'd also like to say that while model organisms are useful for understanding the generality of most biological processes, they are not perfect, right? And to move from that imperfection, what I suggest that with the genetic and phenotypic tools that are rapidly emerging, we can begin moving toward making organisms models for themselves. And I think that would be beneficial because we can make direct comparisons of the direct changes that can occur to an organism and study them in that way. And it also makes for more work for more researchers and more universities to hire good professionals to complete within their institutions. With that being said, I would like to close with some acknowledgements. I would like to acknowledge my dissertation committee who definitely helped me kind of center my research in a way that made sense to everyone, including my advisor, Richard Lenski. I'd also like to thank my collaborators, including two wonderful undergraduate students that I had the opportunity to work with while at Michigan State University. A good friend of mine, Dr. Rohan Mansetti, and a close collaborator on many of my projects, the Lenski Lab in some. I'd also like to thank several institutional places here at MSU, that was at MSU, Beacon, and NFF Center for the Study of Evolution in Action, the Microbiology and Molecular Genetics Program, the Michigan State University Summer Research Opportunities Program, from which I gathered this very talented undergraduate student, Ali Abdel Majid, and also the Ecology Evolution and Behavior Program. Unfortunately, I can't take any questions. This talk, if you were there at my live talk, my internet froze, and that's because I am in Idaho and sometimes these mountains are not forgiving. So if you have any questions, feel free to email me, hit me up on Twitter. I'm always available. Ciao.
So our next speaker is Carlos Garcia-Robledo from the University of Connecticut. And he will be telling us about work he's done in the rolled leaf beetle system, asking about the resilience of insect populations to projected global warming. So I'll pass it off to you now, Carlos.
Hello, can you guys hear me?
Yep, and the slides look good.
Excellent. All right, Megan, thank you so much for this invitation. This is so exciting that actually we made it after all what happened, right? This is super, super exciting. All right, everybody. So today, I want us to go visit some tropical mountains. And I'm going to be talking about tropical mountain passes. I'm going to be talking about population dynamics in insects and how those population dynamics will be affected by climate change. Okay, so we all know that we are losing species at an unprecedented rate. And by 2050, we might lose something like between 15 and 35% of species that will be at risk of extinction. And the main generators of these extinctions are habitat loss, species invasion, overhunting, climate change. And climate change is connected to habitat loss by the loss of habitat along tropical mountains. So here we have an example of a typical tropical mountain in different ecosystems. So with an increase between 2 to 6 Celsius degrees in the next century, what is going to happen is this, right? You're going to have a shift of ecosystems uphill. You're going to have new temperatures in the lowlands and potential extinctions. But for sure, something that is going to happen on top of the mountains is that those habitats will be reduced and maybe they will disappear. So it is assumed that tropical insects are more prone to extinction than temperate species on the predicted global warming because their physiologies are adapted to tropical, more stable temperatures. But are tropical insects really more endangered by global warming than temperate species? Okay, so here we have a classic paper that was published in the American Naturalists, why mountain passes are higher in the tropics. And the general idea is that thermal niches and fitness of tropical insects are adapted to local temperatures that are more stable in the tropics. And what do we know about how ectotherm responds along these latitudinal gradients? So this is a classic paper that actually explored this by measuring fitness. And I really love this paper. And the whole idea of this paper is that they use life table analysis that estimate fitness, and then they plotted for tropical species the relative fitness at different temperatures, and they did the same for tropical and temperate insects. And the general result is that the fitness of tropical insects increases at lower temperatures, but actually the fitness of temperate species is higher at higher temperatures. And this is the figure, the central figure of this paper, where they projected changes in fitness for tropical and temperate species in the year 2100, and the fitness of tropical insects is supposed to decrease. So I was really interested about this data set, and I actually contacted the PNAS office to track down the original data sets, and this is what I discovered. All tropical species, insects included in global analysis, are either crop pests with broad geographic distributions, or biotic agents, usually biocontrol agents, usually raised in the laboratory for multiple generations. So in conclusion, we know so little. We are actually trying to understand fitness of tropical insects by using 9 species raised in the laboratory, when we have, in the last estimate of one publication in our laboratory, we estimate that maybe we can have 5 million, 8 million, maybe 20 million of species in the tropic. So if we want to really understand fitness of insects along latitudinal and altitudinal gradients, what we need to do is to actually visit different localities and measure fitness at different elevations and latitude. And for this, we need inexpensive technology to let us measure these differences in vital rates and fitness at a broad geographic scale. So how can we instantaneously visit all these different elevations and latitudes? So it would be amazing if we could have a device where you can just get in, and then instantaneously you can be at any elevation and latitude, and then you can actually measure fitness in that locality. And I don't know if you guys are thinking the same that I'm thinking about, right? This device exists, well, at least in our imagination, right? So this is the TARDIS. So if you remember Doctor Who, he was able to travel anywhere and in any time in the galaxy using this device, the TARDIS, right? The time and relative dimension in space device. And actually we have TARDI, but TARDI are super expensive. We can have incubators, we can have growth chambers that can mimic the environments of anywhere on Earth, but they are super expensive. So the idea would be to have devices that can actually simulate these environments, these tardi, but make them super cheap. And then we can replicate these studies at different elevations and latitudes. And that's what we are doing. So let me introduce you to our project that I call the $500 Laboratory of Interactions and Global Change. And the objective of this project is to design accurate and inexpensive equipment to estimate thermal limits and demographic rates of ectotherms at different elevations and latitudes. And the whole idea of this project is that we have these workshops where we bring like parts to build equipment. We discuss with students what do we want to measure, some thermal limits, demographic rates. Then we build equipment, and then the equipment stays in the place where we perform the workshop. So here are some examples of devices from these workshops. We have this device that costs like $20 to measure upper thermal limits, a mini fridge that can be built also with super like available parts available anywhere. That is a mini fridge that you can bring to the field to measure the lower thermal limits. This is an Arduino controller that can actually be attached to any fridge, and then you have an incubator that is as accurate as the ones that you can, like the commercial incubators that you buy for the laboratory. Here is an example of a device that we are using now in the field to mimic global warming in raw leaves and other micro habitats. And here are some examples of publications from these workshops. And if you want to know more about this, please visit our GitHub to look at some of our designs. And we have already some tardi running. We have one here at the University of Connecticut, one with my collaborators in Mexico, the one in Costa Rica that I will be talking about, and also colleagues in Barro Colorado Island in Panama are working on building their own tardi, and they're starting now. But for today, I'm going to talk about one of our study sites, that is the La Selva and Braulio Carrillo National Park in Costa Rica. And this is also called the Barba Transect. And this is the highest elevational gradient of continuous forest in Central America that goes from tropical rainforest up to mountain forest. And along this gradient, we're studying different interactions. And the core system in our lab are plants from the order Zingiberales. So these are the banana-like plants. And we study, for example, the insect herbivores that feed on these plants. We also study mites that hitchhike on these beetles, and they need to use the beetles to move from plant to plant. We work on some projects on gut bacteria, the pollinators. We have here a hummingbird visiting a flower, but also this group of mites that feed on nectar and hitchhike on the beaks of hummingbirds to move from plant to plant. And also we have collaborators working on the plant chemistry and the association of this chemistry of the plants with plant herbivore interactions. So, but for today, I'm going to talk about what I think is the core system in my laboratory that I started working with since I was a grad student. And well, this sort of, it can be, I can say that is more or less the love of my lifetime in the natural world. And these are beetles from the genus Cephaloleia that feed on plants from the order Zingiberales that include the Heliconias, Costaceae, Zingiberales, Canaceae, Marantaceae, the banana-like plants, right? And these Cephaloleia beetles feed inside the scroll formed by the young leaves. So you have these young leaves that are rolled, and the beetles feed inside those rolled leaves. And they are, for this reason, they are also called the roll leaf beetles. So these beetles, if you unfurl one of those roll leaves in the field, in the neotropics, this is what you will see. You will see multiple species of beetles feeding inside the scroll of that roll leaf. And then when those roll leaves mature and unfurl and expand, roll leaf beetles have to fly. They have to look for another host plant, and they have to colonize this new roll leaf to start the life cycle. So for this interaction to become a model system, I started working on this since my dissertation to gather the following information. First, we need the elevational distributions of plants and insect herbivores. We need the plant herbivore associations, but also we need to know in some level the strength of each interaction. For example, how much each beetle species feed on each different host plant, right? This is a map of our elevational gradient in Costa Rica, the Barba Transect. At the base is La Selva Biological Station, where we have our laboratory. And in blue are the different shelters along the elevational gradient. And the first thing that I did for this project was to trace the elevational distribution of all the host plants and all the beetle species in this tropical mountain. And of course, bam, this is a tropical elevational gradient. So you have this huge diversity. So the main challenge was to understand the diets of all these beetles, who is what, but also how their diets change along this elevational gradient. So for this, what we developed were techniques based on DNA barcodes. There are short sequences used to identify species. And what we did was to put together a DNA barcode host library where we had DNA sequences from all the potential host plants for this virus. Now we developed techniques where you can extract DNA, plant DNA from the gut contents of the virus. You can match it with the DNA barcode library, and then you get something like that, right? You have all the different interactions between insect herbivores and plants. And the final result, this is an example of one of the elevational gradients where we work. We have actually the elevational distribution of each virus species, the elevational distribution of each host plant, but the different colors represent actually how much each virus species fits on each host plant along this elevational gradient. So from this system, we learned already that virus species seem to be adapted to local temperatures along the elevational gradients, and high elevation species have lower thermal limits than species that are in the lowlands who have higher thermal limits. But this is only based on insect physiology. And we all know that the temperature at which insects faint is not fitness. So if we really want to understand the responses of insects to global warming, what we need to do is to actually understand the population dynamics. So what we have here is the fundamental equation of life history, that is the kernel for every single demographic model that we use now. And the fundamental equation of life history says that if we know how long do you live, survivorship, how many times do you reproduce in your lifetime, then you can estimate fitness. And I want you to look in this equation at this red circle and that parameter, that is the parameter we're going to be talking about today. That is little r. So who is little r? Little r is the instantaneous population growth rate. It's the parameter that links ecology and evolution. And little r is fitness of individuals in a given environment. So how do you measure fitness? Here we have an example of a cohort raised in the laboratory. And then we have individuals. Each line represents an individual. The one here on the top is an individual that was born and then survived for 1 day as a larva and died. Here we have another individual who was born as a larva, survived for 3 days, pupated, then here turned into an adult, and then died. And then if you follow the schedule of birth and death of that cohort, and you organize them from the individual who lived the shortest to the longest, you have this curve on top. And that's the survivorship curve. But in this case, the survivorship curve, LX is not the equation. It's actually empirical. Okay, and what we have here is that after you have these adults, you need to follow their schedule of fecundity, how many eggs and larvae they produce. And if you follow that schedule, you have this curve, and that's the fecundity function. But in this case, it's also an empirical function. And if you can combine LX, survivorship, with MX, then you can calculate little r, that is fitness, the instantaneous population growth rate. And we need to remember that fitness always happens in an environment. So this is our laboratory at La Selva Biological Station, and every single container that you see there has one individual beetle from different species feeding on different diets, and they are raised at different temperatures, and we're measuring LX, survivorship, and MX, fecundity, to measure fitness along this elevational gradient. And here is also a representation of the elevational distribution of all these different beetle species in this tropical mountain. But for today's talk, I'm going to talk only about one beetle species, this one that you see here. This is Cephaloleia beltai, that is the roll-leaf beetle with the known broadest elevational distribution in this tropical mountain. And we know that Cephaloleia beltai has two different mitochondrial haplotypes. You have a lowland mitochondrial haplotype, then you have also a high elevation mitochondrial haplotype, and you have a hybridization zone. So we know that these two haplotypes are actually one species because you can produce in the laboratory hybrids that are viable. So for the first question that I want to explore in this talk is, are high and low elevation haplotypes adapted to local temperatures? So for this, what we did was to follow the birth and death and fecundity schedules for over 1,000 individuals for over 3 years. And using this information, we were able to estimate fitness of individuals in a given temperature. So we have the elevational distributions of the different haplotypes, so you can map them by sequencing part of the mitochondria. And also we have a long-term data set of temperature along this elevational gradient. We have data for each elevation collected for 4 years, every half an hour. And using those data, we have the average temperature at each elevation, we have the minimum temperature and the maximum temperature. And we use that information to inform the temperature treatments that we use in the laboratory. So for the first experiment that I'm going to talk about today, we obtained from males and females mated from each haplotype an F1. And then we raise this F1 at these different temperature treatments. And these temperature treatments can be used to map fitness of these insects along the elevational gradient because they represent the average, minimum, and maximum temperatures at each elevation. So for example, if you want to estimate fitness at the highest elevation, the average temperature at the highest elevation of the mountain is 15 Celsius degrees, the lowest temperature is 10, and the maximum temperature is 20. So if you use these combinations, you can actually use these 5 treatments to assign different temperatures during the development to environmental temperatures. So are high and low elevation haplotypes adapted to local temperatures? So what I'm going to show you now are survivorship curves. And these are survivorship curves for the larvae of the high elevation and low elevation haplotypes. And what we have here at the bottom is a summary of the main results for each one of these figures. So here we see that for the high elevation haplotype, larvae survival is higher at 15 and 20 Celsius degrees, that is the temperature on top of the mountain and the middle of the mountain. But larval survival of the low elevation haplotype is higher at 20 and 25 Celsius degrees, that are the temperatures in the middle of the mountain and the bottom of the mountain. So larval survival seems to be adapted to local temperatures. These are survivorship curves for adults, so this is adult longevity. The high elevation haplotype is also adapted or has higher longevity at temperatures associated with high and middle elevation, whereas the low elevation haplotype has higher longevity, the adults, at temperatures that are typical of the middle of the mountain and the lowlands. And now fecundity, we have the same pattern. So fecundity for high elevation haplotype, the high elevation haplotype is higher at temperatures that are typical at high and middle elevation, whereas the fecundity of the low elevation haplotype is higher at temperatures that are typical of the middle of the mountain and the low elevation. So fecundity is also seems to be adapted to local temperatures. Okay, and if we combine survivorship with fecundity, we can measure fitness. So here we have estimates of Darwinian fitness for the high elevation haplotype and the low elevation haplotype. And the main result is that the high elevation haplotype is adapted to cold temperatures. Population growth only happens at temperatures between 15 and 20 Celsius degrees, but the low elevation haplotype only displays population growth at temperature between 20 and 25 Celsius degrees. Okay, so are high and low elevation haplotypes pre-adapted to future warming conditions? So for this, what we did was that we have this data set of temperatures recorded at each elevation every half an hour for 4 years. So what we did was to connect the estimates of fitness for each haplotype with environmental temperatures along the elevational gradient to estimate mean fitness. And these are the results that I'm going to show now. So here we have for the low elevation haplotype that is adapted to warm temperatures, we have population growth decline ratios and also have the average projected fitness. And then at the bottom, what we have here is 0 represents current temperatures. And then what we did was to simulate temperature increases from 1 to 2 to 3, 4, 5, 6 Celsius degrees of climate change in the original data set to estimate what is the effect of increasing temperatures on fitness. So here we have the first result for the low elevation haplotype. And we see that, as expected, you have population growth in the lowlands, and also you have population growth at middle elevation, but you have population decline on top of the mountain, because this haplotype is adapted to warm temperatures. If you increase temperature 1 Celsius degree in the data set, you see that you have a little bit of population decline in the lowlands, but also instantaneous population growth rate increase at mid elevation. And this is the result. At the end, if you increase temperature 2 Celsius degrees in the simulations, in the lowlands, the low elevation haplotype will suffer demographic attritions. Here are the results for the high elevation haplotype. And what happens here is that you have the opposite pattern, right? You have that you have population decline after 2 Celsius degrees are increased at mid elevation. But the summary here is that in the low range of the distributions, you have demographic attritions and extinctions with a little bit of temperature increase. But in the higher range of the distributions, actually you have still population growth for both populations. So both haplotypes will become locally extinct at the bottom of the ranges with an increase in temperature beyond 2 Celsius degrees. High elevation ranges are thermal refugia to global warming. So I talk about this hybridization zone along this elevational gradient for this beetle species. So the next question that I want to answer is if are hybrids adapted to current temperatures? So for this, I have 2 different hypotheses. The first hypothesis, I call it the hot mitochondrial hypothesis, and it is, and the idea of this hypothesis is that the thermal limits are inherited through the mitochondria. So that means that your thermal tolerance will depend on who is your mother. So if this is true, if the mother of a hybrid is a female from high elevation, then I would expect the hybrids with high elevation mitochondria will survive at high elevation, but not at low elevation. And if the mother is a female from lower elevation, I would expect that hybrids from this female will be able to survive at high elevation, at low elevation and hot temperatures, but not at high elevation. And maybe they can persist in the hybridization zone. And then my second hypothesis is that actually when you decouple the mitochondrial genome from the nuclear genome, then you have this maladaptive genomic decoupling effect, where when you have hybrids actually are not able to expand to colder or warmer temperatures. And basically what is happening is that they are trapped in the middle of the mountain. They are trapped in the hybridization zone. So we did the following experiment. We collected females from high elevation, so cold tolerant, and the circle represents the mitochondrial genome, and the bar represents the nuclear genome. And then we have low elevation individuals, they are heat tolerant. And again, you have hot mitochondria, and then you have nuclear genomes from low elevations. Then we obtained a hybrid F1, where we had individuals with mitochondria from high elevation and mitochondria from low elevation, and then the nucleus is heterozygote. And then what we did was that we distributed the hybrids in different temperature treatments. So 15, 20, and 25 Celsius degrees to test if they're able to survive or not at these different temperatures. So here we have similar results as I showed before, in the same format. We have larval survival for the hybrids, and the larvae of the 2 different hybrids were able to survive at temperatures that are equivalent to the temperatures at mid elevation. For the adults, adult longevity is higher at temperatures that are equivalent to the top temperatures at the top of the mountain and the middle of the mountain. And then fecundities are higher at temperatures that are equivalent to temperatures in the middle of the mountain and in the lowlands. So what I'm going to do now is I'm going to summarize how these vital rates for each one of these different haplotypes and hybrids are distributed along the elevational gradient. So this is an example of the data that I'm going to show you in the next slides. So we have an example of 2 temperatures, and then this is an example where vital rates are coupled. They're adapted to a given environment. So you have coupled vital rates where larval survival, adult survival, and fecundity are all adapted to the same environment. Here we have another example where actually these vital rates are decoupled. So in this case, in this example, you have the larvae survive better in temperature 2, but adults are higher longevity and higher fecundity in temperature 1. So let's see what happened, right? So here we have the results summarized for the different elevations: low elevation, mid elevation, high elevation. And then we have the result for the haplotypes high elevation, haplotype, low elevation, and the hybrids. So as we saw before, the haplotype, the high elevation haplotype, both larvae, pupae, and larvae, adult, and fecundaries are adapted to high elevation temperatures. As we saw before, low elevation haplotypes, larvae, adult, and fecundaries are adapted to temperatures at mid elevation and low elevation. But what happened with the hybrids? Here you have that those vital rates are decoupled. So larval stages have higher survival at temperatures equivalent to the ones at the middle elevation. Adults' survival increases at temperatures that are equivalent to the middle elevation and high elevation, and then fecundaries are higher at temperatures equivalent to the mid elevation and low elevation temperatures. And for the second hybrid, we have exactly the same pattern. So far, we are still measuring fitness for these hybrids in the laboratory, but this evidence can only suggest that maybe the mating genomic, maladaptive genomic decoupling hypothesis is what is happening, right? So basically, when you decouple the mitochondrial from the nuclear genome, it seems that those hybrids are maladaptive when they are exposed to extreme temperatures and they are trapped in the middle of the mountain. So
we have 3 minutes left.
Excellent. So in conclusion, I want to go back to why mountain passes are higher in the tropics, the classic paper by Jansen. And I wish Jansen was here listening to my talk, because I think that in many ways, these results are a textbook example of his hypothesis. The mountain passes hypothesis predicts that temperature stability along tropical mountains promotes local adaptation and speciation. Mountain passes are even higher in the tropics than we previously thought. Not only species, but in this case, populations are adapted to temperatures along tropical mountains. And mountain passes are especially high in hybridization zones, and hybrids are maladaptive. Also, we saw in our simulations that an increase in temperature of just 2 Celsius degrees will trigger demographic attritions, pushing species and population boundaries to the limits of tropical mountain passes. Thank you very much.
Great. Thank you very much, Carlos. We've got a few questions here in the chat. I think we have time for just a couple, so I'll get going. We have a question: how different are the mitochondrial DNA haplotypes from each other? And this question refers to their genomic differences.
Well, so what we have in the data set right now is we have, this is the gene CO1. So we have thousands of DNA barcodes for the beetles too, right? And for this particular species, the difference is tiny. It is from the 600 base pairs, it can be one quarter, if that, of difference, not even that. So when you, let me see, the percent of difference between CO1 for the two haplotypes is something like 1%. That's the answer, if I remember well, or less.
All right, I'm going to try and squeeze in this next one because I had the same question. Does your climate temperature model include the possibility of increased variability that comes along with climate change warming?
Yeah, so that's interesting, because the issue is that we're using this approach where we have fixed temperatures for the treatments, because that makes feasible to actually have data sets for multiple species. And these are dozens of individuals that we are trying to track, right? If you're trying to mimic the actual temperatures at each elevation, so you have the fluctuations, right, during the day and during the night, then the experiment becomes super complex, because you have to figure out what is an average temperature for each hour, right? Or what is if you start a cohort at a given time, when are you going to introduce into experiment? Because you're going to have different peaks of temperature, right? So the answer is no, right? We have these models where we have fixed temperature, and then what we do is that we actually simulate with these piecewise functions the normal reaction of fitness amongst different temperatures, and we assume that actually that's simulating fluctuations.
Great. I think that's all we have time for, but there are more in the chat.
I think, yes. Great talk, Carlos. And yeah, unfortunately, we have to cut off the questions. So we will move on. The next speaker will be André Green, who will talk about how he's developed the Monarch butterfly system, or uses of the Monarch butterfly system for studying Eco-Evo-Devo. So with that, I'll turn it over to you, André.
Awesome. Are we set to go?
Yep, looks good.
Awesome, great. Well, first of all, I want to thank Meg again for giving me the opportunity to kind of be in this lineup with everyone. The talks have been super interesting so far. And kind of like Meg said in the introduction, I work with a system that's actually pretty familiar to a lot of people. But what I kind of wanted to talk with you about today was how I've been thinking about how to use this system in a kind of relatively different way from how it's been kind of traditionally thought about. Monarchs are a big system for thinking about behavioral ecology or population dynamics or even infectious disease dynamics. But I've been thinking about monarchs in the terms of thinking about complex traits and how they evolve, and thinking about how we can potentially use monarchs to answer a relatively old question and thinking about the evolution of migration. So let's jump into this. Awesome. So there are these really, and we're thinking about model organisms. There's these classical systems of studying the genomic basis of adaptation in natural populations and thinking about how particular adaptive traits can evolve really rapidly. So there's heliconius mimicry systems, there's kind of coloration and behavioral changes in cichlid fishes, and the sticklebacks, there's skeletal changes, behavioral changes as well, and then of course Darwin's finches, beak size, and additional morphological features. And what these systems have kind of allowed us to learn is that, first of all, adaptive traits can be due to kind of a range of different genetic architectures, from very specific mutations that lead to trait change to highly polygenic systems that lead to differences between either populations or species. But also studying genomic bases of change has also revealed sometimes really surprising ways that species have interacted in the past. For instance, the heliconia system has really kind of pinpointed this idea that adaptive introgression being really important to kind of how mimicry has evolved across these butterflies. And also, in kind of zooming out, I guess, a little bit, thinking about the genomic architecture of how rapid trait change may happen might provide some insight into how species potentially will respond to rapid climate change. And I'll see if we can think about that a little bit towards the end of this talk with respect to monarchs. So in the systems that I just highlighted, I wanted to think about how we can extend those systems. I mean, we've already kind of learned so much, but there's certainly kind of ways to go. And two kind of ways I've been thinking about this are, first, in the systems that we looked at, we were looking kind of at either adaptive radiations of traits or the convergence of traits. So, but what's also kind of prominent and particularly rapid trait evolution is the fact that there's fluctuating evolution. So in this kind of little model here, you can go from this blue phenotype to orange, but then switch back to kind of this original phenotype. And this actually is pretty prevalent. And second of all, how do we start to incorporate the fact that environments are not static and that they're dynamic throughout time? So one complex trait that people have been thinking about the evolution of it for quite a long time that incorporates dynamic environment and repeatable evolution are animal migrations. So just three kind of quick examples here of New World songbirds, salmoniid fishes, and temperate zone bats. What you tend to find is that migratory status and other migratory traits will actually switch relatively rapidly across some specific clade or set of species, shown by kind of the different colors at these terminal edges. Now, there's already a pretty robust framework for trying to understand the evolution of migration, and it's based in Wright's threshold model. So in this particular figure, we're looking at the frequency of individuals on the y-axis and what's called the liability trait. And in this case, this was a model describing the Eurasian blackcap, and the liability trait for these blackcaps is migratory activity. So in this model, there's some particular threshold over which you will be a kind of migratory bird. And if you don't reach the threshold, you will be a resident or a non-migratory bird. And what's important here in this model, the liability, migratory activity, is being polygenically controlled. So this x-axis is also kind of indicating genetic variation for the liability. So given this kind of quantitative genetic model, thinking about lability or evolutionary lability of migration actually becomes straightforward. The idea being that you can select for, there's genetic variation to select for residency, i.e. to the left of the threshold. But if you then, selective pressure then starts to select for migration, there is quantitative genetic variation that's available to be selected upon, and you can basically kind of shift to the other side of the threshold, i.e. selecting for migration. So if you basically oscillate across the threshold, that leads to kind of our model of evolutionary lability. But thinking about this, well, this model of migration evolution a bit more, complexity actually frames the quantitative genetic model. And what I mean by that is there's an expectation for how difficult or easy it is to evolve migration in the first place, essentially. And the expectation has been that because migration is such a complicated trait, it's integrating behavioral characteristics with physiology, with morphology. Because of this complexity, it should be relatively hard to evolve de novo, which has been kind of the more widely accepted model for the evolution of migration. But it has also been posited in the past that because migration, first of all, evolves so quickly and it can look quite different, or some aspects of migration can look quite different in different lineages, that this might be an indication that migration actually evolves pretty frequently, evolves de novo pretty frequently. And given a rare versus frequent de novo model, there are some different expectations for what you'd expect if you were looking at the genetic basis of variation in migration, or if you were looking at, for instance, the distribution of those species that do show migration. In a rare model, you might expect kind of the same or parallel genetic changes across different migratory lineages, and expect some phylogenetic clustering. Whereas in a frequent model, the genetic changes will more likely be convergent, and there should be a more random distribution of those lineages. And of course, this is all controlling for different ecological factors like habitat, for instance. I mean, because of course that's going to kind of be your ultimate control of migrate or not migrate. But what I'm interested is in thinking about, are there genetic architectures that either promote or constrain the evolution of migration? So in the past 10 years or so, migration, or the evolution of migration, has gotten this infusion from comparative genomics and population genomics. So lots of migratory species now are having their entire genome sequenced. And what this has basically led to are some kind of complications of our expectations from the quantitative genetic model and the rare and frequent de novo scenarios for complexity. And basically there's data that's supporting kind of everything. For instance, in the Eurasian blackcap and willow warblers, what you find instead of the polygenic basis for these traits, you find lots of large effect loci. Whereas instead of finding the same or parallel genetic changes, if you look among relatively closely related species, for instance, if we now add this, for instance, thrush, if you look at the loci that are predicted to determine migratory status or traits in these different systems, there's no kind of initial kind of suggestion of functional relatedness among those loci. And they're certainly not in overlapping parts of these different lineages' genomes, which is what you would have expected of the rare model. But then you also migratory lineages aren't kind of randomly distributed across a particular clade. There is some clustering, which kind of then leads back to suggestion of rare de novo evolution. So I guess kind of putting it all together, if one can even do that, is I think about this more of a hybrid model, in that there is some type of kind of ancestral evolution of a central migration apparatus. And that actually kind of potentiates losing or regaining evolution. And that can happen through diverse genetic mechanisms. So kind of the two primary questions that leads me to think about are how exactly does migration evolve de novo, and kind of what allows for rapid transitions? And I think both of those questions can be addressed by thinking about the fact that migrations are actually strongly kind of integrative traits, and they've been proposed to kind of function as the so-called migratory syndrome. And what I mean by that is that although there are kind of very kind of specific aspects to migrations and specific lineages, all animal migrations have kind of this essential structure to how they function, i.e. they take some environmental input. Behavior, physiology, and morphology are integrated in a particular way that kind of yields a migration output, migration phenotype. So I think the question then to thinking about how does, and each of these traits, I should point out, each of these traits on their own, behavioral traits, physiological traits, and morphological traits, they're not specific to migration necessarily. It's the fact that they're combined in a particular kind of spatiotemporal setting that kind of culminates in a migration phenotype. But you can separate each of them and they function in different kind of parts of the organism's overall function. So then the kind of key that makes a migrant or makes migration is what exactly is that specific integration of behavior, physiology, and then kind of these additional either morphological traits, et cetera. Because of that integration, if you select on kind of a specific portion or one of them, then the correlation between the traits means that you can achieve kind of more rapid evolution of the correlated traits. So this is addressing kind of that rapid evolution component that we want to think about. And now trying to think about de novo, how does migration arise? And my own background is in Evo-Devo. Evo-Devo, kind of one of the kind of main points of the field is thinking about how do novel traits arise. So this is kind of the framework in which I think about this problem. So we come to monarch migration and how we can kind of fit monarchs into this model of migration evolution, and kind of really focusing on this Evo-Devo component while being allowed to integrate this with quantitative genetics and population genomics that are all possible in the monarch system. And we'll get to a little bit of that. Okay. So some background on monarchs. First of all, monarch migration is a result of phenotypic plasticity. So in spring and summer, each of these dots in these graphs was basically a citizen scientist reporting to the Journey North website that, hey, I've seen an adult monarch. So over spring and summer, monarchs are all across the US and southern Canada and Mexico, as you'll see. And there are also monarchs, we are going to focus primarily on these that are east of the Rockies, but there are also monarchs that are west of the Rockies. So these individuals are reproductive throughout spring and summer, in about, well, actually 2 to 4, 5 is pretty rare, overlapping generations. They are primarily looking for food and mates, and they will live about 5 to 7 weeks, and their females will lay eggs on the host plant milkweed of different species. But in fall is kind of the dramatic transformation, in the sense that all of these monarchs that used to be kind of across the US and Canada will, and these are single individuals that will basically halt their reproductive development at what's called reproductive diapause, and they will fly southward, basically all congregating at these very specific sites here in central Mexico. And that's what kind of these aggregations will look like. This is kind of a zoomed in view of monarchs on a branch. And this is kind of the zoomed out view of a piece of the forest in Michoacan. Monarchs will spend their winters there in the subsequent spring. They will mate in Mexico, and the same individuals will begin to fly back northward with the reappearance of milkweed kind of in the southern part of their range, and that will kind of repeat the cycle. So these monarchs, instead of living 5 to 7 weeks, can live up to 10 months. Okay. So I focused my description on kind of the more common population. This is the Eastern North American population. But it turns out that monarchs are actually relatively kind of diverse, even just in North America. So in this figure here, we've described kind of this Eastern flyway. I briefly mentioned that there are monarchs that are west of the Rockies that will migrate to the central and southern California coast and northern Mexican Pacific coasts. But in addition to those migratory populations, there are some non-migratory populations even in North America. One of the most prominent being in southern Florida. There is a population of monarchs that will not migrate at all. They are resident and they will reproduce year-round because there is a species of milkweed called tropical milkweed that lasts year-round. And there are also, where these question marks are, populations in northern Mexico that are that are thought to also reproduce year-round, because there are potentially some species of milkweed that can, again, persist throughout the year. These have been much less well characterized, but there have been several kind of reports of them, so we're sure that kind of they're there. We just don't know much about them. The interesting thing about North American monarchs broadly is that they're almost completely genetically indistinguishable. The Florida population actually does show a distinct genetic signature from any of the migratory monarchs, and we haven't sequenced any of these from northern Mexico. But what that suggests then are that the phenotypic differences that we see here are a result of plastic differences. Okay. But monarchs aren't just in the US, they are actually a global species, spread across 5 different continents, several different island systems. And they basically got there in these 3 different kind of dispersal events, to kind of one to the south into central and northern, the northern part of South America, and these kind of red dots, one across the Atlantic, so in Spain, Portugal, and Northern Africa, and one across the Pacific, so Hawaii, Southern Pacific Islands, and then they've made it to Australia and New Zealand. Now, the vast majority of these populations will not migrate. They're resident populations, but some of them, we've talked about the North American migrants. There are migrants in, particularly Australia has a really prominent migration, although it's much more akin to the Western migration than the Eastern migration in terms of its scale. But it's thought to be more than kind of simply seasonal directed movements and to be kind of an actual migration. New Zealand is, some describe it as a full migration. Sometimes it's described as kind of more directional movement. But it is a relatively kind of, it's one of the stronger examples of, after Australia, of an actual kind of migratory movement. And we know that among kind of all of these populations, there is actually pretty strong genetic divergence. And a few years ago, 101 monarch genomes were sequenced, and a kind of pretty strong population genomics report came from that. And what it showed was that there's, as I mentioned, strong kind of genetic differentiation, and it's spread throughout the genome. So there are a little more than 500 protein coding genes that are within loci that are strongly divergent between migratory and resident populations. And on the left side, this is kind of confirming that North American migratory populations are basically kind of genetically indistinguishable, but you start to see differences even once you move from Hawaii and then to New Zealand. So one of the things that we need to understand here, and we're kind of only in the beginning of this work, of my lab and maybe 1 or 2 others, is that we need to understand kind of the basis of these phenotypic differences among these different populations, because it's still left open whether it is kind of, are we describing kind of the genetic capability to migrate, or are we describing plastic differences due to the environment that you're currently in? But there is preliminary but convincing, potentially convincing evidence that in the case from going from the United States to monarchs colonizing Hawaii, and then a subsequent colonization of Australia, that this may actually represent an evolutionary reversal. And this would provide kind of our great system for testing kind of the genetic basis of how do you de novo evolve migration. So what exactly are we doing? We're measuring behavior, physiology, and morphology. So behavior is measured in what's called the well, it's currently measured in what's called the Moritz and Frost flight simulator, where the monarchs are tethered and they get to kind of spin in 360 degrees. In summer, these bugs will either not choose a direction or they'll spin around in the apparatus, but in fall, they'll orient, and this is in North America, they'll orient in a south or southwestward orientation. And that's our indication that there's some this specific directional behavior that they have in fall. We'll measure reproductive traits in females, that's mature oocyte count, in order to determine diapause status. And we can also look at morphological differences. Migrants tend to be bigger, they tend to have more elongated wings. That's proposed to be more efficient for the long flight that they have to make. So
you have about 3 minutes left.
Oh, okay, so we're gonna run through some stuff. Okay, so one of the interesting questions that kind of I've been really fixated on is the fact that we haven't been able to induce migration in the lab. Although we know about kind of day length, temperature, and host plant being these qualities that change from summer and fall, they aren't, while they can induce diapause, they aren't sufficient for directional flight behavior. So I'm interested in experiments that will basically take an unbiased view on what potential cues are being paid attention to in order to switch to migration. And we're taking kind of a developmental transcriptome approach for that. And what we're going to be looking at in those studies is trying to figure out what is potentially coordinating behavior and physiology. And there are suggestions from previous work that either it potentially could be epigenetic mechanisms, hormones, or cis-regulatory elements that may control that integration. And we have the functional genetic tools available to us to be able to kind of answer those questions pretty precisely. So what this all kind of culminates in is this model that I've kind of, my working model for now, and how migration evolution may work in monarchs, and the fact that in environment 1 or summer, there's initial kind of non-migratory population that, due to the integration of behavior, physiology, morphology, responds in such a way that yields migrants in environment 2 in fall. And it's because of seasonal plasticity that cryptic genetic variation is maintained in the population. And that variation then provides the substrate for rapidly evolving migration de novo. So that gets back to how do we see migration evolve quickly? So I think I have to leave it there. I'm sorry that that my timing was a little off, but I'm definitely happy to kind of take any questions.
Thank you, André. I don't know, Nick, it might be a little short for questions. What do you think?
Yeah, my clock's reading 1:30.
Oh, okay. All right.
There are questions in the chat, which you can look at.
My 4th grader has my phone, so I can't check that anymore. Okay, so we will move on. There are a few questions over in the chat. So next up will be Swanne Gordon from Washington University in St. Louis, who will tell us about her work on her non-model model system, the aposematic wood tiger moth, and really interesting work she's done to help understand why populations show variation in warning signals. So let's see. Swanne, I'll pass it off to you now. I actually, oh, there you are. Okay, now you've popped up.
Okay, let's actually get the presentation. Can you all see that?
Yep, that looks good.
Awesome. Okay, get this out of the way. All right, well, thank you so much for that introduction. And I want to first start by thanking you, Professor Duffy, and the other organizers of this particular symposium for inviting me today, and also ASN Assimilar 2021 for giving me and these other brilliant co-speakers the platform to highlight our research and our thoughts today. Okay, there we go. So a central goal of evolutionary biology over the past few decades has been to understand the origin and maintenance of trait polymorphisms in nature. And now in that respect, color represents one of the best examples of functionally and important polymorphisms, probably owing to the fact that they're quite tractable, but also quite widespread across nature, some of these examples that I've listed here. So I have two primary research systems. And in both, a major focus of my work, which includes a lot of field biology, is to examine the benefits and maintenance of diversity in nature. So today in this short talk, I want to highlight my lesser known system, the aposematic wood tiger moth, as an upcoming model system in the topic of color polymorphisms and aposematisms in general in nature. And so for the past few years, my colleagues and I, led by the brilliant Johanna Mapes, has been asking the particular question, why do aposematic wood tiger moths come in so many different colors? Now, aposematism was coined by Poulton in 1890, and basically is a combination of a warning signal with some sort of unprofitability, generally chemical. Now, it's problematic in both its origin as well as its maintenance. For its origin, one can imagine that educating those naive predators were probably bad for those early educators, as the schematic indicates of the monarch butterfly, so nicely following André's talk. The blue jay goes ahead and eats the butterfly, it gets sick, so it learns after a while, you know, I don't want to eat anything that looks like that particular form, that organism. Great for the blue jays to learn efficiently over time. Great for the monarchs in general, but really bad for those early educators. So how did it become established? It's also puzzling in terms of its maintenance, because theory supposes that aposematism is expected to lead to monomorphism for efficient predator learning via positive frequency dependent selection. However, despite this prediction, there are numerous examples of polymorphisms across a variety of aposematic systems, which in my opinion provides us the wonderful opportunity to understand and examine these selective factors and forces behind diversity in these more complex cases. So wood tiger moths, are they toxic? I love showing this video because it's super cute. Well, maybe not for the moth, but this is a video of a bird. He's attacking a moth. The moth has two lines of chemical defense. They have an anal defense that targets specifically insects, such as ants. And then they have a neck gland defense made of pyrazines that as soon as they are attacked by predators, such as birds, they can kind of release this neck fluid and the bird will taste it and just like shake its head in disgust. It just hates it. It'll wipe its beak clean. It generally will drop the moth. And we know from pilot studies and experiments that a moth can actually survive an attack of this sort. So really, really cool line of defense. Wood tiger moths are variable in coloration. They're chemically defended, like I said, and they're widely distributed throughout their life cycle. So their larvae are aposematic, and you can see that the color is variable. The warning signal can be really bright and big, and it can also be quite small. They overwinter as larvae. They'll come out in the spring. They'll start eating again, turn into adults. They have a short flying season, about 2 weeks to a month. And then the female will lay a couple hundred eggs and start the life cycle all over again. The females also have continuous coloration or variable coloration from yellow to red. And then the males, for instance, in Finland, the males have discrete coloration. So in Finland, they have two morphs, yellow and white hindwing male. Generally, the wings are folded, and so the males look quite similar when they're resting. But when bothered by predators or anything else, or when courting females, they open up their wings and then they show these brilliantly colored hindwings. Some populations in Finland have an admixture of both morphs, whereas others have just one. But what's actually kind of interesting is when you look across their global range, their global polymorphism. And then you see some really cool features. So the ones here, these are the males up in the top with all their discrete morphs. And down here, this panel are the female distribution across their range. You can see this area here with the red for both males and females. Our molecular assessments have shown us that these are kind of the ancestral region or the range for these guys. And then as they spread across the globe, then you see the establishment of other morphs, both in the males and the females, some of them going to fixation, and other places, these morphs kind of coexist with each other. So, sorry about that. So again, we expect positive frequency dependent selection to be working in this case, with predators avoiding the most common warning signals in each locality. But although we would assume that in all of the experiments that we did in this system, we wanted to actually confirm the presence of it on a global scale. And we did that in a really big experiment a few years ago, just published this year, well, this past year. So we exposed morphs to local predators across 4 different countries, as I'm showing you here, and we tested whether selection by predators favors the locally common morph. We also monitored the abundance and community structure of birds in each locality to test whether or not this community structure of their predators are associated with attack risk to these different morphs. By doing this experiment, we provided the best documented case to date, in my opinion, of a geographic mosaic of selection on warning signals at a broad spatial scale. So again, we did it in 4 study locations ranging from monomorphic populations to polymorphic overlap of these morph forms. We used artificial plasticine moths with printed wings. And if you want to find out how we did that, you can go and look at any of these 2 papers. We did it across 60 predation transects where we randomly placed 20 white, yellow, and red moths. We left them for 6 days during the flight season, recorded their predation events daily, and then we also recorded the bird community again at each site. So these are our results, and this wonderful study was mainly led by our PhD student, Katja Ronka. And so it's kind of busy, so I'm going to run you through it slowly. So up top, we have our local morph frequencies. Again, we did this in 4 different countries, and even though we can put all of the data and the work into this picture right here, it was actually a lot of work that went into it. And so Scotland is monomorphic for the yellow form. Georgia is mainly monomorphic for the red form, and then Estonia and Finland are polymorphic populations with different ratios of white, yellow, and the red form. Below, we have our expected predation risk if positive frequency dependent survival selection is probably in each of these localities. So this is the attack risk from low to high. Again, Scotland with its yellow monomorphic population, we would expect that the yellow morph would have some kind of advantage against predators. Same for the red morph in Georgia, and then the combination depending on their frequency in Estonia and Finland. Below, we have our actual results. So these are the attack risk across Scotland, Georgia, Estonia, and Finland. Altogether, we observed a total of 718 bird attacks on the 4,004 artificial moths that we placed out. And our results were as we expected. So in Scotland, the yellow morph has an advantage. In Georgia, the red morph, which is the most locally common morph, had an advantage. And in Estonia, the white morph had an advantage, although you can see that the predation pressure is quite low in Estonia compared to someplace like Scotland, which is probably why you don't see that huge variety. And then in Finland, the yellow morph, which was the least frequent morph, had the highest attack risk. Okay, so our results on the predator community actually alerted us to another cool issue. So not only did we find positive frequency dependent survival selection across this global scale, but when we grouped the bird community by feeding tactic to describe them and then linked it to morph survival, we found these really cool stuff. And so this is the estimated risk attack for each of the morphs. And then here's the principal component loading the different bird predators. On one end, we have tits, great tits, blue tits, and on the other end, we have the dunnocks and warblers. But what was really interesting, so just looking at Finland, is we found that in Finland, the yellow morph is favored in communities that are more heavily characterized by tits, whereas the white morph is more heavily favored in communities characterized by dunnocks, as you can see from the higher attack risk to the other form. In contrast, in Scotland, that's mainly monomorphic yellow, you have this different type of result, this opposite result. So really, really interesting that the predator community is actually quite important, right, to these morph ratios and attack risk probabilities to each of these different forms. And it really wanted me to focus, or led me to focus more specifically on one end, one extreme of this figure. So looking at great tit behavior that might actually be quite important. But it also alerted us to another really cool issue. So in 2014, Ossi Nakalainen and others found that the yellow males in southern Finland were attacked significantly less than white males. Whereas in our study in southern Finland in the same sites, we found the opposite result, right? We found that the yellow morphs were attacked more. They didn't have an advantage over the white morph. So we were puzzled by that at first because we were like, well, it makes sense based on what we thought from the morph ratios. But then we went back to our censuses that we had been doing in these populations over 10 years. And it alerted us to the idea that the frequencies of yellow and white morphs varies in southern Finland in a biannual cycle. And the yellow morph was more common during the Nakalainen et al. Study, whereas the white morph was more common during our experiment, again, providing more evidence into, even though it's such a short spatial scale or temporal scale, that you still have the signal of frequency being important. Okay, so this is just a kind of picture of our censuses that we collect over time. So this proportion of yellow males, either fully yellow or fully white, across the different years, showing you how it kind of fluctuates in southern Finland, whereas in other places like central Finland, it's actually quite stable. Now, many studies in frequency-dependent selection in aposematic systems focus quite broadly, as we did in our previous study, and they may miss information on the actual predators, their behavior, and variations in their responses that might provide mechanistic insight as to the role that frequency really plays in warning systems. So I wanted to really focus in on the predators and using a more stable local population, such as central Finland, and then including the defense, because before we've mostly been using these plasticine forms to kind of get rid of that aspect. So we decided to do an experiment that kind of thinks about things like temporal fluctuations, so looking at a smaller, finer scale, and then also the predators themselves. So we did an experiment where we used recently freeze-thawed moths, recently killed moths, so we could have their defense without the variation of their behaviors. Let's see. So we did it in an indoor enclosure with a one-way mirror, so we could see the bird predators, but they couldn't see us. We had 4 treatments. So we had a balanced control where we had equal numbers of each of the forms. We had a white bias treatment. So we had 12 white moths compared to 6 yellow and red forms. We had a yellow bias treatment and then also a red bias treatment. We tracked the wild predator, which was a great tit in our case behavior. So their beak cleaning and also their ability to attack quickly, to taste, and then to drop promptly the moth, which are both indications of distaste in a bird. We also recorded the age of the bird. So we had some juveniles and then some adults and their orders of attack. The experiment ran until the bird ate half or until of the moths, or until 2 hours had amassed. Okay, so did we find, is there positive frequency dependent survival selection on this shorter spatial and temporal scale? So looking first at the control, up here again is the attack risk, and the control where we had a balanced ratio of all 3 morphs, we found no significant differences between them. In the female bias treatment, again, we found no significant differences between them. But when we got to the white bias treatment, we did find as expected. And so when the more common morph in the treatment ended up having a survival advantage over the other forms. So just as predicted by theory and our prior wider scale studies. When we looked at the yellow morph, we did not find a significant difference. However, there is a trend for the yellows to have a survival advantage over them. And so for the white morph, it was quite strong, possibly expected because the population in central Finland, as you probably saw from my past figure, is quite heavily white biased. But when we analyzed the data in whole, we found that the red morph also is under positive frequency dependent survival selection. There's a benefit to be in common, but only when you consider them in comparison to their much lower survival when they're rare. So looking at the white biased and the yellow biased, when the females are at the lowest number, so only 6 individuals out of the 24 that we have, then you see a much higher feeding rate against them compared to when they have higher numbers. Okay, so this was really, really cool results. And, we wanted to get it out right away. But we also looked at characteristics about the predators, and we wanted to see what other characteristics about the predators or the moths might underlie these results. So this is just looking at the probability of beak cleaning and then the probability of promptly attacking and then dropping the moth. So basically the results show that the red morph elicits more beak cleaning, which again is a toxic response, while the white morph elicits more grabbing the moth, tasting it, and then dropping it right away. Maybe the yellow morph, which we didn't really see that much of a response to when the predators would attack and eat them, relies more heavily on behavioral differences, which we did not account for, of course, in our study. So to save time for the rest of the talk, you can ask questions in the chat or ask me later what I think have solicited these differences between the morphs and the treatments, and also more specifically about bird age and sex, which we found also affects attack risk in really cool ways. So in mini summary, in general, the theory is correct. Whites suffer lower predation when common, positive frequency dependent selection. Reds cause higher hesitation and also suffer higher predation when rare. And positive frequency dependent selection we found in this small study, again, is quite important, but likely complicated by predator type, age, sex, and variable responses to prey profitability, showing that the selective outcomes in these types of experiments might actually be more dynamic than we think. So they highlight the need to understand predator foraging in natural communities when we're thinking about studies such as this. So positive frequency dependent selection should lead to fixation, which is what I told you in the beginning. So what does this mean for a global polymorphism, right? If we're looking across the range of the geographic mosaic in the system, how is polymorphism maintained in the face of positive survival selection, frequency selection? The short answer is that we don't know fully yet. And in each of our papers, we pretend that we do, and we're working on it. However, a past experiment that I did may hold some clue. And so an earlier study of mine in a large outdoor enclosure tested whether negative frequency dependent selection in the context of sexual selection could potentially balance the morph ratios and allow for rare morphs to flourish, as expected. So here we conducted an experiment under semi-natural conditions where we estimated mating success under 3 different morph frequencies in central Finland. And this time the red morph, which are mainly females, were used in the treatment for, the mating. And you can see these 2 corresponding papers if you want to hear more, because I'm pressed for time. So basically we expected negative frequency dependent reproductive selection, but what we actually found was positive frequency dependent reproductive selection. So it kind of puzzled us to have 2 signals of positive frequency dependent selection. Then you would expect to find across our global range the fixation in all of these places of one specific form. So we next integrated our data from the mating experiment with known estimates of predation against both morphs in a simple spatial model in collaboration with the brilliant Hannah Coco. And this trade-off was particularly interesting and underexplored in the case where both natural and sexually selection, again, are potentially frequency dependent in aposematic organisms, as never before had variation in frequency dependence across fitness components been considered in this context. So please go read the paper and go ahead and cite it if you want to. And so you don't have to run through all of this. I don't have to explain it too much because I just want to get to the results, which showed us that when we include kind of the spatial dynamics of there being environments where the predator community might confer an advantage to one morph versus in another environment where they have an advantage to that other morph, combined with the two aspects of positive frequency dependent selection and a little bit of gene flow or dispersal between the different subpopulations, then you can find that polymorphism can be maintained in this system. It doesn't explain everything that we see across the range, but it does give a nice answer as to what exactly we're seeing. So our broader conclusions: positive frequency dependent selection in both mating and survival combined with dispersal can lead to polymorphism in this fantastic system. Multiple factors contribute to diversity, with many more needing to be studied. And we have a lot more important papers to come in the next few months. So, watch out for them. And with that, I want to end by thanking my funding sources, collaborators, my mentors, and people that give me advice. And then, of course, I recently opened my own lab last year at Washington University in St. Louis. So come join us. We're always looking for excited people. And I'd like to thank also my future, my babies, my two children who make me the happiest person alive. And ever since this summer, where, everything was happening in the US and the world, and I'd been given these platforms to give these talks, quite often over the past few years. But ever since this summer, I decided that in addition to thanking my future, I also wanted to make sure that every talk I thanked my past. So the many named and nameless heroes that have paved the way for me in education. And since this summer, again, it's been really important that I highlight their sacrifice and their resiliency. Because of that, I stand in front of you today, proudly and unapologetically Black in STEM. And for some, that statement may be triggering, but I think the triggering point should be the vast amount of data that we have amassed already, such that Professor Duffy has highlighted, in her introduction this morning, that we as scientists should listen to, showing us time and time again the stark racial disparities that we are seeing within academia, in publications, in hiring, in retention, in funding, and most importantly, in opportunities. Now, how long before we as a group decide to break down the barriers and the academic gatekeeping, right? Before we open our eyes and see, but most importantly, react to the biases that run rampant in our institutions. Now, the world is going through its own reckoning right now, as many people have brought up, but that, especially the US where I am right now, but that gives us no excuse not to also act here in academia. And I'm starting to see the beginnings of this, and I'm so proud of it, but we still have a ways to go. Because like my research shows in Nature, and like many of our research shows, diversity in every facet is our strength and not a weakness. And with that, I want to leave you with two quotes that, I absolutely love. One is by a prominent Canadian researcher taken too soon in his time. And then a second is by a writer that is continuously inspiring to me and other people. And with that, if we have any time, I will take any questions or comments. And if not, then I will be sure to answer them in the chat.
Well, thank you, Swanne. That was a great talk and an apt ending there. So we have a bunch of questions coming in the chat, so I'll just get to it. Becky asks, why should there be negative frequency dependent sexual selection if there's strong positive frequency dependent survival? I would think that adaptive mate choice would favor positive frequency dependent mate choice. Is it inbreeding avoidance?
Yeah, that's a great question. I mean, when you think about the theory behind positive frequency dependent survival selection, of course, the theory points heavily towards it going to fixation, right? Towards there being one form. And something that we know in terms of polymorphisms and things that can maintain polymorphisms in nature, generally, one of the main things is negative frequency dependent selection, right? It's something that balances, that allows those rare forms to establish themselves or to flourish or to proliferate afterwards. And that's why we went to kind of look at that first. So that's why I went for it. I mean, I'm coming from the guppy system where negative frequency dependent selection is huge in that system, and a reason why we have such a broad variety of body coloration in that specific system. So when I came to the wood tiger moths, of course, it was the first thing that I was going to go to. Why is it that we have these balanced polymorphisms in places? Why do we have some places where it's highly monomorphic, but then right next door, you have like a local population where all of these morphs kind of coexist, almost at, you know, for the males, for instance, in some parts in Europe where there's only two, at equal ratios. And so the first thing for me to look at was negative frequency dependent selection. But in addition, we're also looking at these other things, such as inbreeding avoidance and things like that. But in terms of balancing that ratio, that was the first thing that we wanted to examine. Well, I did when I joined the lab, the group.
So another question here, how do empirical measurements of dispersal compare with your spatial model results?
Yeah, so that's one of the fun, cool papers that we have coming up next, is actually looking at dispersal in these guys. We've, of course, looked at it molecularly. And so there was a brilliant paper published in 2015 by Robert Hegna and colleagues that's kind of looking at the biogeography of these guys. So when we look at this broad spatial scale, we know that these guys have traveled far, right? We know that they've gone many places. We know that they're rare and hard to find in some places, but we also know that when we would collect them, that we'd have a hard time, say, recollecting them, so that they potentially move quite often. But again, we didn't have a finer assessment of this ability. And so I ran for a couple of years intensive mark recapture in one of the sites in Finland, where we were able to actually collect the fish, collect the moths over the flying season to actually get estimates of dispersal in these guys, both morph-specific dispersal and also population-specific dispersal. So we do know that dispersal is actually very high. It's a high rate for these guys, more than we even expected. And, you know, we combine that with molecular assessments over the years. So trying to look at turnovers between them. And we find some really cool things. I don't want to give it all away because I'm hoping that I want to give a couple talks on this soon. But we found some interesting things, such as, for instance, the females that we know do not move that much because they're, of course, filled with eggs, that they have a much lower dispersal rate. And so as we're looking at relatedness over the different years, we can see that in the females, it's a lot stronger, a lot higher than, for instance, the males, which we know move actually quite far. And so that's coming up. We're looking into it, and it matches wonderfully for our model, but it kind of pushes the boundaries because, of course, our model needed dispersal to be kind of small. And we're seeing that they actually disperse a lot, at least the males. And so that's definitely something that we want to expand later in any future models or experiments.
So I'm going to try and squeeze in at least one more. Do the moths co-occur with other, either toxic or not, moths? And does that have any relationship with the polymorphism and predator responses?
Yeah, so that's a brilliant question. And I'd like to point you again to some research done by Katja Ronka, who's a graduate student in the lab and now is a postdoc. And so she wanted to actually explore that idea of generalization, that potentially in certain environments such as Georgia, where it's the only place really that you find a lot of those red male morphs, that it could potentially be because there are red colored other species that are quite that are very toxic, right, compared to our guys, that might actually be aiding their establishment in Georgia rather than in the other places where they have spread to. So we do know that on a smaller scale, just looking at our local birds in Finland, they don't seem to generalize that much between the morphs. We do not see a strong sense of our results being driven by alternative prey or alternative species in the system. And we also did that in the large scale study that I just told you about, in that we took a census of all of the alternative prey and butterflies and moth species in our transect and included that in our analyses, but we do not see a strong pointing to that on a broader scale. But it is something that we're definitely continuing to look at, and that data was not included in that paper, and I'm sure Katya is hoping to expand on that a little bit later.
Well, thank you again. I think that's all we have time for.
Yes, I think we have to move on, but that was all very interesting. Okay, so our last speaker in this symposium, I'm so sad it's ending because I'm loving these talks. Our last speaker is Rachel Penczykowski, who is also at Washington University in St. Louis, though it's a coincidence that we have two speakers from the same place. And Rachel will be talking about how Plantago species can be used as model systems. And it's getting noisy here, so just take it away, Rachel.
Great, thank you so much, Meg. It's such a treat to be part of this really excellent symposium. Okay, am I in correct presenter view here? All right, great. So I'm, like Meg said, going to tell you about Plantago species and their usefulness as models for studying both plant herbivore and plant pathogen interactions across environmental gradients. So the questions of how and why biodiversity change along environmental gradients have been asked in ecology and evolutionary biology for a really, really long time. And of course, there are many different types of abiotic factors that vary not just discretely in terms of a high, low, yes, no form, but continuously or mostly continuously. And so if we imagine a hypothetical abiotic gradient here shown in this red to blue color scale, we might see a pattern in nature, a pattern of biodiversity, where these different shapes could represent different species or different functional groups or different genotypes within a species. And so then we'd want to know why this pattern has been formed and what maintains it. And there are, of course, both ecological processes that can lead to these types of diversity patterns over gradients. And these would include the big high-level processes that Wale talks about, so speciation, dispersal, drift, and selection among species. At the same time, there can also be evolutionary processes that produce such patterns, including the sort of parallel processes of mapping onto the ecological processes on the left. In the evolutionary processes, we have mutation, gene flow, drift, and selection within species. And so the relative extent to which these different processes are operating and producing these patterns is going to depend a lot on what the pattern is. What is the spatial scale that you're examining? What's the temporal scale and the history of that site? What is the dimension of biodiversity under consideration? Is it functional, phylogenetic, or genetic within species? And what are the types of organisms? What's their mode of reproduction? What's their dispersal ability and other relevant traits? So many different things to consider, but I'll start with a relatively straightforward example of an abiotic factor occurring as a gradient at a few different types of spatial scales using readily available temperature data from the World Clim database. So we see this quite apparent dramatic pattern of mean temperature change with latitude. Here I'm plotting January temperatures because that's where we are, January. But we could also look within a continent, say, at elevational patterns, such as in the Andes in South America, where we see this green band along the Pacific coast much cooler temperatures at the higher elevations. So these are patterns that explorers have noticed since the dawn of exploration. But more recently, we're made aware that temperature and other variables also change quite dramatically over much smaller scales due to processes that humans have created, including processes involved in urbanization. And so you might not know what this map is showing, but for reference, this is the city of St. Louis, bounded by the outline there, and downtown St. Louis, the Gateway Arch at that eastern edge of the city. St. Louis County is actually made of 88 different municipalities and 10 unincorporated municipalities. And the WashU Tyson Research Center is just on the western side of St. Louis County. And so even in this region where I am currently, and in Swanne as well, we see this really stark gradient in temperature, where the downtown area that's more built with human structures is a lot warmer, especially in summer months. And the surrounding suburbs and then rural areas are much cooler. So these are, of course, just one example of an abiotic gradient. And an example of a pattern of biodiversity that would map onto such a gradient at a global scale would be the dramatic gradient in biodiversity of several different types of taxonomic groups. This map shown here is showing the species richness of extant vertebrates globally, and we notice these striking hotspots of biodiversity in the tropics. Elevationally, there are also really interesting changes in community composition and biodiversity. Shown here is this classic map that Humboldt and Bonpland published, showing the distribution of plants on Mount Chimborazo in Ecuador. And so these types of changes in the distribution of organisms with latitude, or here with elevation, and what types of abiotic factors they map onto, what types of other types of biotic patterns they map onto, has long fascinated ecologists and evolutionary biologists and naturalists. And so, the question of why is species richness greater in tropics and at lower elevations for many groups? Of course, there are exceptions, notable exceptions to the rule, but it is the case that for many groups, there is this pattern of increased diversity towards the tropics and at lower elevations. And there are numerous non-mutually exclusive hypotheses about the historical, ecological, and evolutionary processes that create and maintain those biodiversity patterns. And so if we now take this hypothetical gradient and say that the arrow pointing to the left is moving us to lower elevation and closer to the equator, a very pervasive hypothesis in our fields of ecology and evolution is that there would be stronger, more specialized biotic interactions at those lower elevations and lower latitudes, and that this could involve stronger selection pressures between species, leading to faster diversification, so creating new diversity, as well as stronger negative density dependence, especially among antagonistic interactions like with pathogens or predators or herbivores, leading to greater, so with specific pathogens or herbivores leading to greater species coexistence. And again, these are not mutually exclusive, and there also are plenty of exceptions to the rule, and much left to be tested about the why of these biodiversity patterns at the among species scale. But then there's also the question of how and why does intraspecific diversity change along environmental gradients? And this is relatively less studied compared to those big dramatic patterns of among species diversity. But there are, people have been considering this question for some time now. And so if we again consider this gradient of a hypothetical abiotic factor, and then examine some hypothetical intraspecific genetic diversity upon that gradient, we would need to consider how factor processes of mutation, gene flow, drift, and selection by both abiotic and biotic factors generate and maintain that genetic diversity, and then how that genetic diversity combines with whatever phenotypic plasticity can happen in that system under this environmental gradient to lead to the produced pattern of intraspecific trait variation that we can then measure. And so there's different approaches for getting at these, testing these different processes and their effects on either genetic diversity as measured through whatever is your favorite approach for measuring genetic diversity, or the trait variation in whatever traits of interest you see to measure. And then ideally, you would put those together and have a more comprehensive understanding of how the environmental gradient and all that goes along with it leads to both genotypic and phenotypic changes. Of course, to many of us, at least speaking for myself, I get especially excited about ways that the abiotic and biotic factors select on species and create diversity in this way. And there have been many recent studies looking at intraspecific variation, especially in anti-herbivore defenses of plants along environmental gradients. And so I just want to highlight a few of these. Latitudinally, there have been a handful of studies looking at and finding declines in the intensity of herbivory and or the level of defense against herbivores and plant species. At least one recent study, probably more, looking at longitudinal variation in abiotic factors and herbivory. There are some, of course, studies looking at elevational gradients. I just had picked one here, but there are others. And then a few newer studies looking at urbanization gradients, including in white clover and the cyanogenesis defense against herbivores. And so there is some research out there, but there are still many open questions about intraspecific variation, even just in plants, considering one relatively limited set of organisms in this world along environmental gradients. And so I want to highlight a few of those open questions here. So there's still much to learn about the spatial and temporal scale dependence of the different processes that create and maintain diversity, and differences in scale both within a given type of gradient, whether it's a temperature gradient strictly, or a latitudinal gradient, or an elevational gradient, urbanization, moisture gradient, you name it. There are many of those gradients can be found at different scales. Then also comparing between the different types of gradients. So for example, further investigating the extent to which an elevational gradient is or is not a good proxy for a latitudinal gradient, or whether or not a latitudinal or elevational gradient with a temperature climb is or is not a good proxy for temporal climate change in a region. Of course, to complicate things further, typically there's not just one abiotic factor changing on its own in a given gradient, but multiple factors. And so you need to be aware of what types of factors are either correlated positively or negatively, or not at all. And plants may also be under selection for multiple types of species interactions at once. I highlighted a few recent papers about plant-herbivore interactions across gradients, but it's the case that intraspecific variation and plant-pathogen and plant mutualist interactions are much less well studied. So some very big open questions comparing between different types of antagonistic interactions like plant-pathogen and plant-herbivore, and then comparing between plant mutualist and plant antagonist types of interactions and how those vary. Another important open question that's been highlighted in some recent papers is that when studying variation in plant defenses, whether against herbivores or pathogens, one needs to consider how variation in resources, so spatial and temporal variation in resources, and growth traits of the hosts confer tolerance that mediates the fitness impacts. So the traits that we measure are really the net effect of how the plant is resisting and or tolerating their enemies. And so there are many other interesting open questions about plants and why they may vary in their diversity across gradients. But for any of these questions, one needs to think carefully about what plant taxa would be useful for studying these open questions. And here I'm talking specifically about studying the intraspecific variation. And so, of course, to study something over a gradient, you would want to find a species or a set of species that are widespread in their distribution across those different types of environmental gradients in nature. You'd want to work with an organism or organisms that are tractable for both field and greenhouse-based studies. You'd want the ability to propagate these species, ideally with high success, by seed and possibly also by vegetative reproduction to clone a single genotype line. And it would also be helpful if there was an existing body of literature on some of the basics of the biology, chemistry. I guess I wrote chemistry twice. Chemistry is really important, and species interactions and what's known for those species. And I think another point to highlight is that, if our goal is to fill existing gaps in knowledge, to pick a species or group of species that are really complementary to other current existing model plant taxa. So of course, Arabidopsis is a very, the most well-known true model plant system for its very useful genetic tools. But you might want to then not choose, a kind of ephemeral annual weedy plant like Arabidopsis if searching for a new type of model organism, and you might want to, search in a different plant family. And similarly, white clover is another really useful model plant species, including for plant herbivore studies and in gradients, including latitude, elevational, and urbanizational gradients. But white clover is a legume. If you want to expand our overall knowledge and fill gaps, then maybe choose something that's not a legume. I'm leading to somewhere here. And then I think another really great feature of a model system is one that's really amenable to collaborative approaches, especially if one is planning to study species across very big gradients that may involve a lot of geographic expanse. And also very useful if such species are amenable to educational modules and also public outreach. So I think as scientists, we need to think about how we can use our species as teaching tools and training tools, and also connect people to the biodiversity that is in their own neighborhoods. And so behold, you say Plantago, I say Plantago. Sometimes I switch it up, but the genus Plantago has over 200 species. Most are rosette-forming herbs, such as the photos that are shown here from this Hassemer et al. Paper. There are, some of these 200 species are quite narrowly endemic, and several are cosmopolitan weeds. And so for the rest of my slides today, I'm going to focus on these cosmopolitan weeds, which in their ubiquity are a bit more useful perhaps in thinking about really big environmental gradients to study them in. And specifically, I'll tell you just a little bit about my favorite three Plantago species, the Plantago lanceolata on the left, Plantago major on the right, and Plantago rogalii on the bottom middle. So there's a little bit of interesting differences in their reproductive biology. All three send up these long flower stalks with the inflorescences at the tip. Plantago lanceolata typically sends up this quite long stalk with the inflorescences way up high. Those are self-incompatible with wind-dispersed pollen. Plantago major and Plantago rogalii, you can see the flowering and seed production starts a little closer to the ground, and they're also self-compatible in contrast. And their leaves are a bit wider, in case you didn't notice that fairly obvious morphological difference. So the top two, Plantago lanceolata and Plantago major, are native to Europe and Western Asia, but largely due to European colonizers, have been introduced pretty much all over the world, where they do quite well. And so they're broadly considered cosmopolitan weeds. Plantago lanceolata is an allergen, so it's a bit noxious, but they're not really to the level of noxious that there's any widespread eradication campaigns against them. So they're kind of established all over and pretty much here to stay. Plantago regelii, by contrast, is native to Eastern North America and remains quite restricted in its distribution. So just to show them up close a little bit more, the seeds of Plantago lanceolata on the top there, starting with the flowering, these cute little flowers kind of flower in a ring, starting from the base of the flower stalk upwards, and then the seed stalk produces a whole bunch of little golden colored seeds. On the bottom left photo there is an example of some herbivory and pathogen that I will tell you about again in a few more slides, but that's a common buckeye caterpillar consuming a powdery mildew infected Plantago lanceolata leaf. Plantago major is also native again to Europe and Western Asia. Also gets eaten and infected by things. And you can see the progression of its seeds and increasing maturity in the photograph from left to right. And I wanted to show this close-up photo just to make the point that Plantago major and Plantago regelii actually look quite similar in their growth form, in their leaf shape and color. There are some kind of subtle differences in the base of the petioles of Plantago regelii is often but not always reddish. The converse is usually true. And the fruits, the size and shape of the fruits are a bit more reliable as a way to distinguish them. And so I point this out because I've actually noticed that herbaria records are not necessarily accurate with regards to identification of Plantago major versus Plantago regelii. And it is also a limitation in the use of iNaturalist and other databases for finding them. So to the trained eye, I mean, I could teach you how to distinguish them, I think, pretty reliably. But one needs to be aware of this when using more broadly sourced types of data. Okay, so what do we know about these species already? There's actually a fair amount of published research, especially on Plantago lanceolata and Plantago major. I've just plotted here the top 30 Web of Science topical categories for these three species. There are only 10 papers total published about Plantago regelii or featuring Plantago regelii, which is kind of interesting to me, given that it's a very, very common weed across Eastern North America. In fact, in many places more common than Plantago major, but very understudied across the board. Just to highlight a few of the categories with the most research, of course, plant sciences broadly has a lot of hits, which makes sense. But there are, several hundred papers about Plantago lanceolata that fall under the ecology category, and more than 100 about evolutionary biology. But notice that for Plantago major, the sort of coral colored dot on this graph seems to be relatively understudied in ecology and evolutionary biology, considering that in many ways it's pretty comparably cosmopolitan and ubiquitous compared to Plantago lanceolata. So I think that's quite interesting. But I will note that there's relatively more studies of Plantago major in topics of pharmacology and pharmacy. It's actually been used as a medicinal herb for millennia. And there are some, if you Google Plantago major, you will find mostly hits about poultices that you could make or ways that you can use parts of the plant as different types of remedies. Some of this has been reproduced in reputable scientific journals. Some of it maybe has not, remains to be rigorously tested. So just to highlight some of the herbivores that these species share. There are quite many, including a few specialist lepidopterans. The common buckeye butterfly consumes all 3 species in North America. So the letters on the table are continents, basically. So North America is NA, and that is our local to me specialist lepidopteran. Melatia kingsia, the glandular fritillary butterfly, is a specialist on Plantago lanceolata in Europe. And in fact, that is the focal host plant species and the focal butterfly species that Ilkka Hanski used in his pioneering metapopulation studies in the Åland archipelago of Finland. Arctia plantagenus, the wood tiger moth, also specializes on Plantago lanceolata and will sometimes consume Plantago major as well. And so the thing that's very interesting about these specialist lepidopterans is that they've evolved to deal with, and in some cases, sequester and use to their own advantage the iridoid glycoside compounds that are putative anti-herbivore defenses in Plantago lanceolata in particular. So these chemical compounds, acubin and catechol, do show some anti-herbivore properties against generalist herbivores, but there are specialists that prefer those compounds and then can themselves be protected from predators. There are, some more general herbivores, Spaptera species, so armyworms, leaf miner flies, weevils, sawflies, and of course aphids and thrips. But I think comparing the specialist and generalist Lepidoptera is an especially interesting set of species to consider because of the kind of opposing effects of these putative chemical defenses on these different herbivores. There are several known pathogens of Plantago species, including powdery mildews, other fungal pathogens, and recently discovered viruses. The powdery mildews, however, are by far the most widespread and well-studied pathogens of these species, in part because they are super obvious. So this is a close-up of powdery mildew on Plantago regaliae. Powdery mildews are obligate pleiotropic fungi, so they only grow on living host tissue. And in fact, they only grow on the surface of the leaf. So they don't go systemic, and they very reliably show symptoms of infection. And so that is really conducive to being a good system for doing a lot of field-based estimates of infection, because you can readily see with your eye. There are exactly—
You have just about 2 minutes left.
Okay. There are exactly 2 species of Plantago that infect, 2 species of powdery mildew that infect these Plantago. They have an asexual and sexual part of their life cycle. And in my lab, we're really interested in how herbivores, especially caterpillars and powdery mildews, vary in their intensity and diversity with latitude, and how that intensity and diversity of their pressure on the plants correlates with plant resistance and tolerance to them, as well as, I think, really interesting questions about the crosstalk between those different pathways, both within and between the different enemy types. And a special passion of mine is how winter climate specifically impacts the host-enemy interactions, especially for powdery mildews, which have a very specialized strategy for dealing with winter. We can use iNaturalist to help us identify survey and sampling sites, which again works better in some cases than others, but is generally quite useful both for the plants and the pathogens. And we recently, with a team of engineers, got commissioned the creation of a low-cost platform for precise automated aerial imaging of field plots, which sends a DSLR camera along a grid. And this takes really nice high-res images that show detail of herbivory as well as pathogen symptoms. And so I think that those are really useful approaches for us to actually measure a lot of things over a lot of spatial variation. Just very quickly, we know that there are many ways that ecology and evolution play out in interesting ways in urban systems and along urbanization gradients. And my lab has been exploring that here in St. Louis. And in particular, my graduate students, Quinn Fox and Mahal Bugai, have been performed 2 years of sampling and have some really interesting preliminary findings about how the, sorry, too many animations here, about how the plant pathogen interactions vary along this urbanization gradient, as well as plant phenology itself. Very slow slide to load. And I think I'm probably just out of time now. So I'll just wrap up to say we have more ongoing work with experiments recently completed, and that I think that these Plantago species, you know, they're not, I'm not introducing something that's totally new here. They're already the focus of some really great global scientific networks. And so I guess the point of my talk here is really to introduce you to these species if you don't already know them, and highlight, I think, some of the ways that they should be used in future work to really answer some of these interesting fundamental questions about how interspecific variation in species is linked to environmental gradients. And with that, I will thank lots of people who have helped establish this study system with me, and in particular, my co-author, Drew Sieg, who's a chemical ecologist, and then my postdoc advisor, Annalisa Lina, who introduced me to these plants. Thank you.
Thank you, Rachel. Oh, go ahead, Nick.
I was just going to say that that's our time for now, so people should feel free to leave if they need. But because this is the end of the session, we can take questions for Rachel or for, I suppose, any of our speakers that are still present. I did have a question about kind of this gradient of intraspecific diversity and how it's related to species diversity, right? You know, there's kind of this body of theory that relates evolutionary processes to ecological processes and the diversity that's maintained at each of those scales. And I wonder if there's, you know, if there are any issues in wholesale taking the kind of hypotheses for the tropical diversity or the latitudinal diversity gradient for species, and then just applying that to intraspecific variation.
Yeah, and I think there are. So there have been a couple of papers in the last few years. I think I put them as citations on some of my early slides. I think maybe probably going to cite the wrong one from memory, but they're in there. But people have pointed out in recent years that those same that same theoretical framework may not directly translate, and so we really need to you know, kind of start from scratch and thinking about what actually we expect to be the driving factors in intraspecific diversity and for different types of taxa. So it's I guess starting from scratch was maybe too strong of a word. I would say the development of that theoretical framework is underway, and in the last few years there have been a number of people starting to lay that groundwork, but there's more to be done still, especially I think thinking across different scales.
Thanks. And I guess we should wrap it up so we can continue these conversations either on Slack or in some other way. So back to you, Meg.
Yeah, thanks so much to all the speakers. I really, really enjoyed all those presentations, and unusually for the timing of this, several of the papers have already been accepted and the others are in revision. So hopefully you'll all get to read the complete stories relatively soon in MNAT. So I'll end by thanking all our speakers again, and also many thanks to Mark and Mike and Nick and all the folks who've been putting in a lot of hard work to make this run. It's gone remarkably smoothly. Thank you.